we have been trying to present watershed models that it is a type of things you should have an integrated farm with an understanding among the farmers that you manage with the small water that is flowing to your area, preserve the water from the rainy season, so that at least 2-3 added crops you can do. But the thing is that when food security is an issue, making these farmers to produce other than paddy or the food grain has been a challenge, but we have been trying our best to increase that.
Yeah, so there are many areas less rainfall is really rich.
Yes.
So in those areas, maybe it's better idea from millet,
Exactly.
bajra, okay, and all other crops.
The thing is that millet has been, the millet has been tried in other states previously also. Millets has a big potential because it has practically the demand for it is a demand that is driving international demand that is driving millets also. So farmers have been very it has been a profitable proposition for many farmers in recent year to shift to millets. But the thing is that, sir, we are always in circle with pulses, specifically pulses and oilseeds that they have been the products of demand. Anyway, sir, pulses, oilseeds have put the country in difficulties, and there have been good rewards for better pulses and better oilseeds. But the issue remains: the seeds are not properly, the seeds are not available for pulses and oilseeds in proper form. The way we address paddy or some other crop, we don't address pulses and oilseeds in that way. Anyway, millet has come as a giving added advantage, and it will practically add to this benefit stream of the farmers. And I think I have sort means what we have been practicing. That is the only thing we can say at this point, sir. Anything else? So, thank you, sir. I think.
Thank you so very much, sir. A big hand for Shri T.P. Das, everyone. Briefly presented on the major drives in agricultural development and the vexing issues in agriculture. Never playing a vital role in rural development and agriculture. It was really nice hearing you, sir. Moving ahead, we have a very special speaker with us, Professor Robert Henry. He is talking on the application of genomics to the sustainable use of plant biodiversity for food and energy. Sir is joined. Sir is already with us from the University of Queensland, Australia. Let's give a big hand to welcome Professor Robert Henry.
Thank you very much for the opportunity to talk to you today. I'll begin by sharing my screen. So today I'd like to talk to you about application of genomics to sustainable use of plants for food and energy. The key message is that the analysis of the genetics of plants with genome technology is something that contributes substantially to the biological understandings that are really the key to both plant breeding, but also the use and management of our crops, because we understand their biology better, and the way in which we achieve the attributes that ensure we meet the requirements of our consumers in the market. So I'm going to talk to you about some of the big objectives of my research program, which include capturing plant biodiversity to improve food security. And here we really look at capturing that variation that exists in the wild relatives of crops and bringing it in to the gene pool so that we can have a greater range of crops, but even going beyond that and domesticating completely new plant species so that we've got more secure future for food because we've got a greater range of plants that we can use. And the other big area that genomics contributes to enormously is our ability to reduce climate change by engineering plants to actually replace fossil carbon. There are many things for which we will need carbon into the future, and we need to have a renewable source to replace fossil carbon, and plants are really the only option, and we need to fast track that if we're going to meet our targets for climate change mitigation. Climate change by engineering plants to actually replace fossil carbon. So there are many things for which I'm not sure if we need to have a renewable source to replace fossil carbon. So what I was going to just talk about is the fact that all of the diversity we have in plants can be and is being systematically searched to find the new sources of food that we can capture from the great diversity, for example, of flowering plants that we have. And the technology that's enabling us to do that really is the great advances in genetic technology. Here we depict the cost of sequencing the genome, and this is based on the human genome. And we can see that this has dramatically collapsed over the years. The real key message is that this process is continuing. It's getting cheaper all the time. We've moved from when this was drawn at $1,000 to sequence a human genome, now to more like $100. And we're going to expect within the course of 2023 that the cost of sequencing platforms, some of the long-range sequencing platforms, will decrease more than tenfold. So we're going to see a dramatic revolution in this technology and its ability to contribute to agriculture. We've been tracking some of these developments, particularly as they've accelerated over the last 2 years or so, by comparing all of the available technologies and seeing how we can use that to analyze our crop genetics. What has emerged is that we can actually assemble the whole chromosome now from just the DNA sequence analysis alone. If we go back to 2020, we were quite often getting lots of little pieces that made up the chromosome, and it was a challenge to completely sequence and assemble the genetic sequence of especially complex plants. But that's no longer the case. We can now do this quite quickly, and this revolution means that we can really accelerate the use of DNA-based technology in supporting plant breeding and agriculture more generally. We've written a few reviews on that I'd refer you to get a sense of the way the technology is changing and will change even so dramatically in the course of 2023. I'd like to talk about some of the applications of that to give you a sense of how we see this impacting into the future. And we've been looking at the question of how we reinvent rice. Rice really is and remains one of the really important crops in the world. And we do need to adapt to the changing climate and the changing needs of consumers in this area. And we've been looking widely at the diversity. And if we look at the wild relatives of domesticated Asian rice, they extend from here in Northern Australia up to China and across to India. And there is really quite a lot of diversity there, but it's largely underutilized. We haven't to any great extent exploited that diversity, and we need to begin to do that if we're going to keep pace with changes in climate and changes in demand for food. We look at the genetic relationships between the domesticated rices. We get a family tree that looks a bit like this. We've got probably about 7 different groups of rices that are grown in different regions around the world. And we really had a great conflict of ideas about how we got to this point. How many times did we separately domesticate rice in different countries, in India, in China, in other places? And we've managed to throw some light on that question by looking at the sequence recently of about 3,000 rices that have domesticated rices that have been looked at closely. And we've found that they really fall into just 2 groups. All of the rices suggest that they only have the same maternal genome. Well, this is the case, it's the chloroplast genome. What that tells us basically is a rice has two mothers, but it has many fathers, because we can see many different types of rice around the world. So what's happened, I think, is in the past, in the deep past, there have been two fundamental domestications of rice at two different places. And those rices have been transported and transferred around the world. And in the places where they've been taken, they've interbred with the local rices to actually become almost identical to the rices or the wild rices of those regions. But they still bear that signature of those two original maternal domestications. So I think the statement there really summarizes what we now understand. But what it tells us is there's a great opportunity to begin that process again from other wild sources of diversity, and to produce rices that may have very much the attributes that we want for the future. And so we're looking across all of the rices, not only in Asia, but Africa, Australia, and South America, to find the attributes that we need for the future. In Australia, we have some very divergent species. We have the most divergent of the close relatives of rice, which is a strange thing to say, but we have the most divergent species that are still interfertile with domesticated rice. So they provide the greatest source of genetic variation. And so we're trying to conserve these species and make them available for rice improvement. There's species right across the north and down the east coast. And these species have been consumed, of course, by humans for a very long period of time. These cave paintings in the north, in the areas where we find the rices, predate any agriculture anywhere in the world. So there's been a long period of human use of these, and we are interested in the extent to which humans have altered the genetics of rice without even domesticating. We do find in areas that we see in the wild hybridization happening naturally, what so-called reticulate evolution, where rice evolves by the species being dispersed around the tropics and diverging, but we've now found evidence that they also interbreed, and those rare events of hybridization between the species play a key role in shaping the species that we see today. And it's interesting to see that we can find evidence of that ongoing in the wild, and some of the wild and undisturbed populations that we have in the extreme north of Australia. These represent great opportunities. Here, grain B is a wild rice, whereas E is a domesticated rice. We have the potential to have bigger seeds, and seed size is a major component of grain yield. So we've got very directly opportunities to improve rice performance by accessing the genetics that exists in the wild. These wild rices also have great nutritional attributes in that they have starches that are more slowly digested, and there's the suggestion that they may enable the production of rice that's healthier for communities that consume relatively large amounts of rice. And that's an increasing challenge in modern Asian communities. Another example of this is sorghum. Sorghum was domesticated in Africa, but all of the diversity in the genus is found, or most of it, in Northern Australia, where we have more than 20 of the 23 species of sorghum. And we're now beginning to look at that. We have very diverse types of potential new cereals here that could broaden our genetic base and improve food security. And so we're working on studying the relationships of those in collaboration with the Joint Genome Institute in the US. And we're beginning to understand where we can capture new and important variation for much larger step changes in the genetic improvement of sorghum, which is one of those crops that does cope with warm and dry environments, and so is likely to become a crop of even greater importance in the future. We see great advances not only in the field crops, but also in the horticultural crops. And these species are ones that we're targeting for advanced genomics applications at the moment. These are the 8 key species that are of local economic importance for us, but they're also very important in many cases internationally. We've produced a high quality genome for avocado, which will probably renew interest in genetic improvement of this crop. We've just completed sequencing 11 species of citrus that represent great new sources of diversity for citrus breeding. Citrus faces some major new diseases in the world, and we need to find genetic solutions quickly to be able to retain the levels of citrus production that we've had. And sequencing these genomes in these genetic maps, we're able to find a great diversity of disease resistance genes in the wild. And we know the wild species are resistant to these diseases that are threatening production. And with this knowledge now, we can very readily transfer that resistance into a domesticated citrus and really meet the challenge of these major new diseases. The mango is another one. We're working with people in India and New Delhi on this, but also in other places in the world on different varieties. And in mango, we've been sequencing much of the wild diversity. And what we've discovered remarkably, I think, is that many of the mango wild varieties, wild species, seem to have been subject to interbreeding with domesticated mango. Mango was domesticated a long time ago, and wild and domesticated mangoes have grown together probably for thousands of years. And it's not surprising that we see now evidence that there's been gene flow in both directions. Domesticated mangoes have contributed significantly to many of the species that we find in the wild. And this is going to give us a much better basis on which to undertake genetic improvement of mango. One species I wanted to mention because of its unique contribution is the jojoba. This is a plant that comes from the desert in America. It's very much of interest to people in Saudi Arabia that are supporting us to do this research. And here we sequenced the male and female plant. A small proportion of plants are dioecious, that is, they have a separate male and female plant. But remarkably, no one had sequenced the complete genome of a male and female. And here we did it for the first time, and we found a Y chromosome in the male that was much bigger than the one in the female. We're talking about 10 million bases of genetic code greater in the male. And in humans, the difference between males and females is quite small, maybe 27 genes. We found a much bigger difference between the male and female jojoba plant. This greater sexual dimorphism was not only associated with the different flowers that you get on a male and female, but in different responses to stress. So what this says is male and female plants have been able to evolve separately down different paths to adapt to the stresses of their environment in quite different ways. And this has given us, I think, many more clues, not only about reproductive biology in plants and how we can manipulate that in desirable ways, but about how plants have a range of different ways they can respond to stress. And so much of that is going to be useful, particularly from this species that grows in desert conditions and provides a whole lot of clues about options we can consider in major crops. Coffee is another species that's benefiting enormously from this. We've had the expectation that coffee is under threat in the wild, and changing climates mean that the places where we produce high quality coffee are probably not going to be able to continue to produce that into the future, with critical temperatures at high altitude changing where we get the best coffee. We need to go back to the wild, and there is great diversity there. And the sequencing that we've done in coffee and its wild relatives is providing a platform for us to continue to produce coffee of the quality that consumers want into the future. I think we'll see the same with other major beverage crops like tea. In addition to sequencing, the other big technology that's really important to understand is gene editing. This is a technology that allows us, once we understand what we have, to change it to suit the needs of farmers and consumers. And so we're trying to use this to understand how plants adapt to the environment with our Centre for Plant Success. But we're also trying to understand how things evolved when plants were domesticated, how the networks of genes have been changed by humans, and how we can manipulate that further, or how we can go back and domesticate new things. One of the great examples of this and the importance of this is fragrance in rice. Back up in about 2005, in my lab, we discovered the gene for fragrance in basmati and jasmine rice by having available to us a complete rice genome sequence for the first time. And that revealed that there was a mutation, a loss of function of a gene that led to the fragrant molecule, but that was also associated with a loss of plant stress response. Fragrant plants, by necessity, were more susceptible to drought and to salt stress because of the loss of this biochemical pathway that was associated with fragrance. By knowing that, we're now able to think about deliberately engineering the plant to both be fragrant and to cope with stress. But it's a great example of how human selection and natural selection sometimes are conflicting. But if we understand it, we can address it and get a better outcome for farmers and for plant production. So we're very much focused on characterizing domestication genes in wild relatives and using genome editing. And in rice, we're using that to undertake a de novo domestication of new Ariza species, but to also improve nutritional quality, traits, and disease resistance in rice that's able to be rapidly enhanced. This is going to lead to domestication of new crops. Here's a wild millet that we're pursuing this in, and this species of rice is a target for de novo domestication as well. We've been working through the desert areas of Australia with the Indigenous people and the archaeologists, trying to understand the plants that were consumed by humans. In the tens of thousands of years there's been humans living in those areas, and in many cases where we believe there were large populations, but what we really don't know is what they were eating. We want to learn that because some of those plants might be opportunities for the future. Maybe when we have climates similar to what we had 50,000 years ago when this sort of food production began. So we're looking to capture biodiversity within the Australian flora, from everything from rice to citrus in this list, and many more. And I think the same approach can be taken globally to enriching the diversity of plants we have in agriculture. So the other point I wanted to speak briefly on is the use of plants to replace fossil carbon to fill this critical role in decarbonising the world. And this is how we use plants to produce all those things that we produce from fossil carbon. It's not just transport that we use and fuel that we use oil for. We use a lot of oil, in fact, more oil, to produce products, plastics, chemicals, all those other things, fabrics. And we do need long-term a solution to that if we're going to reduce our reliance on fossil carbon. So this involves fairly significant consideration of how we balance the application of genetics to food security, energy security, and of course, continuing to conserve the biodiversity we need to sustain the environment and to sustain the genetic resources on which agriculture depends. And this is a book I wrote back in 2010, trying to rationalize the challenges that those three objectives create for us. But really, we're interested now in engineering plants to have a biomass composition that allows us to produce biomaterials and things like aviation fuel. The electrification of ground transportation means that we're less likely to need this for cars in the future. Many of us now have electric cars, and many more will have electric cars in the near future. And so we need to focus our efforts with plants on the needs of things like long-distance flight. And here we've got a program to do that, and on the production of things other than fuel that we can't replace in other ways. And the two big targets that we have are sugarcane, and sugarcane is the most produced crop in the world, and eucalypts, which are the most planted trees in the world and representing a woody biomass that's storable. They're two big global opportunities that we're applying genomics to. In sugarcane, we've been working with the sugarcane genome for a very long time, and this is the reason that this species has been such a challenge is that it's the most genetically complex probably of all crop plants. It's the last of the 20 major crops in the world to have its genome completely sequenced. But I can say that we're about to report that. The sequence has been completed, particularly in collaboration, again, with the Joint Genome Institute. And so this genome will be available, and we will be able to say that all of the major crops in the world have have a reference genome we can use to support genomic breeding. We've also developed a whole lot of technologies to manipulate the composition, and one is sorghum mutants, working with the Carlsberg Research Laboratories. With a major program there, we've developed a very large population of mutant sorghums that can be screened to find a mutation in any gene of interest and to test the phenotype. So we're able to systematically now look at every gene in the genome and understand its function. And by doing that in sorghum, a very close relative of sugarcane, we should be able to then translate that genetics into producing the energy crops we need for the future. And this high throughput mutagenesis has been successful to date in finding mutations in every gene we've been looking for very quickly, because we pre-prepare the population and have the DNA available for screening and can find the plant within a matter of days. The same with the eucalypt. These trees are something we've been able to sequence the genomes of and found in the different lineages remarkable processes of parallel evolution. These two groups of eucalypts diverged 65 million years ago, but remarkably have chromosomes with very similar genes. And so we now expect that we'll be able to, in the longer term, manipulate the genomes of these to be ideal replacements for fossil carbon. So I think just by end by thanking all the very many key collaborating laboratories that have been involved in the work I've touched on in this talk, and to thank the many collaborators working in the lab that have done all of that research. Thank you very much.
What we are just replacing nucleotides. So there is no worry for antibiotic. I know from the which thread you are asking this nowadays, we are discussing everywhere. There is no threat. Any other? Yeah.
I have a small suggestion, sir.
Sir.
It will include human health also in genome intervention.
Okay.
Plant genome intervention,
Mhm.
please include human health aspects such as.
It is really great suggestion. And that is the reason, sir, we have now the mission. It is called FUNI security. Food, health, nutrition, energy, and environment security. And I have sent one what is called suggestive letter to Honorable Prime Minister on that area very recently. Yep.
Especially in the fields of, as you told, the, of course, you have mentioned also the diabetes, blood pressure, blockages. These are the main hindrances of human interference,
Yep.
the human activity.
Mhm.
And if you can solve through genome intervention in agriculture,
Mhm.
that will be a great help to the human society, to the world as a whole.
Okay, thank you very much. I will take couple of minutes, particularly from Dr. Bansal, because he is in a hurry. He is to attend another meeting. I share one incident from you, from my past experience. I will share one incident with you. That is when my wife remembers, would definitely remember when Dr. Borlaug was on bed with cancer. Okay, he used to blindly love me. I don't know why. I don't have that credential at all. So he told me, Chitto, enough is enough in molecular evolution and fundamental basic science. You do something which is of immediate need of the society. I asked, sir, what you suggest me? He told, why not cancer? He was suffering from cancer then. Okay. And immediately we wrote one research project, submitted to USDA NIFA program, and without any review, just from concept note, it was approved. So blessings from Dr. Burlog, or this is needed for the society, God knows, but that work we did. Sir, as you know, bitter melon is not only bitter gourd in India, it not only having anti-diabetic principles, it is having also many anti-cancer products. We did some good work on that by molecular breeding, but you are absolutely right. That must be included in genomic designing strategy. Yes, it is a great suggestion. Any other from the seniors? Now I feel comfortable. Okay, thank you very much for your patient hearing. And I thank the organizer again. And I am very thankful to the students of Centurion University. I envy you because you got such a good scope to be in this great campus. Thank you. Thank you, sir.
Thank you so very much, sir, for the great presentation on the reasons and missions of next generation.
If this increase in CO2 concentration, small change in the increased concentration, carbon dioxide. And what about oxygen? Oxygen concentration in blue is anaerobic earth. There's no oxygen. Okay. Around 2.5 billions of years ago, evolved cyanobacteria or blue-green algae, which broke water to form oxygen in the process of photosynthesis. So oxygen concentration in atmosphere increased, and this oxygen atmosphere is photosynthetic oxygen that has increased from 0.01% to today's 20.9%. Okay. And what happened? Methane, the gas were all talked about, dreadful gas were all talked about. Water full of methane, okay, maybe due to geological regions, but today's methane concentration is extremely low because anaerobic life have this. One of the regions being, not only region, one of the regions being the anaerobic life has some substance reduced who are using methane, could emit methane. So methane concentration has substantially gone down through the process of evolution. So we have very low carbon dioxide world, low CO2 world in context of geological past. We have high oxygen concentration world, okay, in relation to geological past, and very low methane in geological past. So if you see our geological past, we had lots of different atmosphere. So climate of the world is changing every year, right from the origin of Earth. Okay, it's not that climate is changing today, but climate changing every year, every day, every minute. Okay, all right. Next slide, please. Okay, if you see that now the temperature. If you see, this is the dotted line, you can see this dotted line is the anything left to this dotted line is cooling phase, anything right to the dotted line is the warming phase. So if you again look at geological past, we are all warm climates except Carboniferous era. Okay, we had 4 ice ages, ice houses before, but a cooling phase. But one cooling phase in recent geological past was Carboniferous era. Okay, you can see this is white here, means the ice house, the cold conditions, the earth. So Carboniferous era had a lot of productivity, lot of photosynthesis, lot of productivity. A lot of biodiversity also took place in the Carboniferous era. And C tree plants flourished in this area, evolved and flourished in the Carboniferous era. Okay? And as we go down in history, you can see Triassic, Jurassic, all is a warming phase, you can see. So all are warming phase. So our geological past is usually warm, usually hot, okay? But in Carboniferous era, we are cold climate. Then came all Cretaceous, Paleozoic, Mesozoic, and now we are actually in the Quaternary. We are in a cooling phase. We are in ice house now. Okay? The geological past, if you see, we are in ice house. We are cooling phase. And what is it? This cooling phase started only 1.75 million years ago. Okay? This is the ice house, cooling phase of the world. Okay. And if you see the sea level, because the cooling phase, the sea level is the minimum today. Okay, to have comparability to our geological past. Next slide, please. Next slide. Yeah, of course, IPCC says CO2 concentration is going to increase. It may reach at the end of century, it may reach 700 ppm, but it may not reach that much. But it may certainly reach 600 ppm by the end of the century, or 650 ppm by the end of the century. And temperature, it may rise by 3 degrees centigrade by end of the century. Why temperature increase, all of you know, because of emission of greenhouse gases. All right. So next slide, please. So we are going to high CO2 world again, going back to high CO2 world again, okay? Because of human interaction with nature. One is CO2 concentration went down due to geological reasons, many other reasons, when we are not there. After Homo sapiens evolved around 1 to 1.5 million of years ago, then because of interaction with nature, okay, we cut down forests, okay, and we, did a lot of, destruction of the biodiversity we created. That's actually led to the increase in the temperature, increase in the carbon dioxide concentration, because mostly due to the loss of the forests. Okay, all right, CO2 concentration, because who absorbs CO2 from atmosphere? The CO2, the plants, the trees. Okay, and CO2, once you cut down the forests, the CO2 concentration is now again going to go up and up. Okay, and it's not only that, the temperature of the earth also is going to increase. Okay. One, of course, so of course the temperature is going up and up and up. Next slide. So okay. So in this context, I want to talk about a plant called photosynthesis. But I told you, the photosynthesis is a process, is the only process on this earth which gives a negative entropy. Okay, reduces the entropy of the world, disorderness of the world, by stitching six carbon dioxide molecules to one carbohydrate molecule. So the randomness of system decreasing once we stitch these carbon dioxide molecules to a single molecule. Okay. So therefore, this is the only process on earth which is creating negative entropy. And another process is it is creating food security for us, providing food for us. And not only that, it's creating oxygen for us. Okay, you can see photosynthetic oxygen has increased. Okay, and can you go back to 2 slides back? 2 slides back. Go back. Back. Yes. You see, say all along were C3 plants. Only 40 million of years ago, okay, you said drop in the CO2 concentration. Okay, 40 million of years ago. When around 40 million years ago, CO2 concentration decreased, okay, so plants became so low, okay, plants were forced to evolve another kind of species called C4 plants, okay, because CO2 concentration in the atmosphere was low, so they had to, compensate for the loss of carbohydrates in the nature for themselves also. So therefore, they had to evolve C4 plants. Okay. So and what happened again? When C4, next slide please. When C4 plants evolved around 40 million of years ago, what's the temperature? Temperature around 40 million of years ago, what you can see here is quite warm. Okay. So C4 plants naturally evolved in low CO2 world and high temperature world. When C3 plants evolve mostly, they evolve in the mostly Carboniferous era, which were mostly cooling phase. Okay. So C4 plants are adapted to high temperature because they simply evolve in high temperature world, and also they evolve in low CO2 world. Okay. Therefore, they can, photosynthesis better at low CO2. Okay. Next slide. Next. Yes. So this is an enzyme called Rubisco, ribulose bisphosphate carboxylase oxygenase, which fixes, carbon dioxide. So ribulose bisphosphate plus carbon dioxide will give you 2 molecules of 3-phosphoglyceric acid, and these are converted later on to carbohydrates. Okay. But the problem is C3 plants, when they evolved, or Rubisco evolved in anaerobic world. Rubisco evolved in anaerobic world. When there was no oxygen, photosynthetic bacteria. Okay, even early cyanobacteria. okay, when there are no oxygen. Okay, so therefore, nature evolved an enzyme called Rubisco, but they did not have an oxygenation function because there is no oxygen there in the atmosphere. So this Rubisco evolved in anaerobic world when there is no oxygen. So it evolved like that for fine, no problem, it objected for millions of years, for billions of years. But when the plants themselves produce oxygen as a byproduct of photosynthesis, then oxygen concentration in the atmosphere increased, and that competed with CO2, okay, to compensate for the loss of photosynthesis. Therefore, oxygenation started when CO2 concentration increased, okay, and oxygen concentration increased and CO2 concentration decreased in the atmosphere. Okay, so it is only happened around 50 millions of years ago. Okay, when CO2 concentration dropped very, 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 very low. Okay, therefore you can see that this instead of forming two molecules of phosphoglyceric acid, is forming one phosphoglyceric acid and one phosphoglycolic acid. Okay, and this phosphoglycolic acid goes to photorespiration, and therefore it cannot utilize in photosynthesis. And therefore, there's a loss of carbohydrate fixed by the plants. Okay. Now, all of you know it. All the students know it. I'm sure. Next slide. Okay. I will not deal with it. I don't have time for this. Next slide. I'm saying how Rubisco evolved and all that. I skip this slide. Yeah. So if Rubisco if you say Rubisco is a very, very sluggish enzyme. So it is like a turtle. Okay. Opposed to that, there is another enzyme called carbonic anhydrase. Okay. Which is very, very fast. It's a cargos. It can learn very fast. Okay. All right. That is carbonic anhydrase. You can see Rubisco very slow. It catalyzes on 3 to 10 reaction seconds per second. Okay. But carbonic anhydrase does 500,000 reactions per second. So carbonic anhydrase is a very high turnover number. Okay. So it can do the reaction very, very fast. Okay. What carbonic anhydrase does? See, water plus carbon dioxide becomes bicarbonate. That's what it does. Okay. But Rubisco does other thing, fixes carbon dioxide, carbohydrate. So Rubisco and carbonic anhydrase are not comparable. Okay. What is comparable is C4 system, phosphoenol pyruvate carboxylase. But I just give an example. Okay. All right. The C3, you can see 50% is Rubisco in a lip protein. And C4 is a little less because it's another PEP carboxylase enzyme. Okay, next slide, please. Okay, I'll skip this slide, don't have to talk about it. Next slide. All of you know this is the rice plant and the maize plant, C3 and C4 plant. Okay, so C3 plants, this is the mesophyll bundle seed cells here, which is, of course, do not have chloroplasts. Okay, maybe rudimentary bundle slit, but here we have a lot of very well-developed bundle seeds having chloroplasts. Okay, so these are C4 plants. The question is, why C4 plant? Next slide. Okay, why C4 plants? Because it is efficient in photosynthesis. Because it does not have photorespiration. Okay, so it has abolished photorespiration in the process of evolution. Okay, and I don't have to talk to details of this. I don't have time for it. Okay, all right. But there's one enzyme, therefore I'm showing you this slide. Plants are through millions of years evolution have evolved an enzyme called phosphoenol pyruvate carboxylase. This phosphoenol pyruvate carboxylase, in addition to Rubisco, ribulose bisphosphate carboxyl oxygenase in chloroplast, there is another enzyme called phosphoenol pyruvate carboxylase. Phosphoenol pyruvate carboxylase is present in the mesophyll cells of the cytoplasm. Okay, and mesophyll cell chloroplast do not have Rubisco. Okay, So plant evolved billions of years, millions of years, enzyme called phosphopyruvate carboxylase. So what is substrate, sir? Phosphopyruvic acid and bicarbonate. There is two substrate of phosphopyruvate carboxylase. So phosphopyruvate carboxylase plus carbon converts phosphopyruvic acid, a 3-carbon compound, plus a bicarbonate, which is a 1-carbon compound, to form a C4 compound called oxaloacetic acid. And the oxaloacetic acid further metabolized later on and again released, increases CO2 concentration in the vicinity of Rubisco to abolish photorespiration in C4 plants. I'm not good details of that. Next slide, please. Next slide. Yeah, next slide. Next slide. Yeah. So what is the advantage of C4 plant? As you said, this is the carbon dioxide concentration on the x-axis, rate of photosynthesis on the y-axis. Okay. So you can see if we, this is 0 CO2 concentration, okay, and we increasing CO2 concentration. Okay. As the CO2 concentration increasing, of course, C3 plants in red. So C3 photosynthesis is going to increasing and remaining constant later on, saturating. But C4 photosynthesis, look at C4 photosynthesis in yellow. At CO2 count very, so because it evolved in low CO2 world, C4 plants, evolved means low CO2 world. So they know to capture CO2 more efficiently, okay, by another enzyme called phosphopyruvate carboxylase, which does not react with oxygen, okay. Therefore, you can see very fast, it saturates very fast, okay, very fast, okay. So today's concentration carbon dioxide is around 400 ppm. You can see C4 plants have already saturated, okay, in photosynthesis. Even if we increase the CO2 concentration, C4 plants are not going to increase their photosynthetic rate anymore. Where C3 plants, photosynthetic rate is still going to go up and may saturate at the C4 level later on. Okay. So you can say therefore how efficiently C4 plants can photosynthesize even at this atmospheric CO2 concentration. Next slide, please. Next slide. Yeah. One important thing of C4 plants, you must say, is that temperature. Okay. C4 plant is a morning to 12 o'clock in the noon, full sun. You can see a C4 plants is doing much better, able to utilize the light intensity, okay, for C3 plants filling. Not only that, compare, this is a very interesting thing, okay. Always C4 plant is not advantageous over C3. In low temperature, C3 plants are much better than C4, okay. As you can see here, at 10 degrees centigrade, okay. Look at the C3 plant. Sorry, C3 plant, okay. So C3 plant and C4 plant. This is C3, this is C4. Okay. So the C4 plant you can see at 10 degrees centigrade is performing much less than the C3 plant. Okay. But 35 degrees centigrade, C4 plants is performing much more than the C3 plant. So since C4 plants evolved in high CO2 world, low CO2 but high temperature world, 40 millions of years ago, therefore they're naturally adapted to high temperature. Okay. So when you're talking about climate resilient agriculture, we should focus on C4 plants for further agriculture research, whether millets or they are any other crop plants, C4 crop plant. That will be our future research, future food, okay? Because at 35 degrees centigrade, you can see the photosynthesis rate has already reduced in C3 plants. And once you go to 38 or 40 degrees centigrade, you'll see sharp decline in the C3 photosynthesis. But C4 photosynthesis, when you go to 38 degrees centigrade, nothing is going to happen to the 40 degree, 39 degrees centigrade, nothing going to happen to C4 crop. Okay, so because they're naturally high temperature resilient. And not only that, they can also conserve a lot of water. Their water conservation capacity is very, very high. Okay, so next slide, please. Okay, I stopped. Go ahead. Yeah, and next. Okay, I will only and a very one or two slides from here, a few slides from here. So what I have done, overexpressed an enzyme called carbonic anhydrase. Okay, as I told you, carbonic anhydrase 20,000 much faster enzyme than Rubisco, of course. So carbonic anhydrase has overexpressed, but we took carbonic anhydrase from Flavaria bidentis. Flavaria bidentis is a C4 plant. It has very, very low Km for CO2. Low Km. Low Km means higher the affinity. C3 plant is around 10 times low Km, means it is 10 times faster enzyme, okay, than the C3 Ca, carbonic anhydrase. C3 plants carbonic anhydrase, C4 also carbonic anhydrase. But C4 carbonic anhydrase has very low Km for carbon dioxide, means a very high affinity for carbon dioxide. Okay, next slide. Next slide. Deepika Kondo did the work. Okay, so there are many kinds of carbonic anhydrase. We will not talk about that. Also in mitochondria, chloroplast, cytoplasm. Next slide. I will skip this slide. Next. Yeah, there are different kinds of carbonic anhydrase present in chloroplast, cytosol, mitochondria, CA1, CA2, CA3, CA4, CA5. Next. Yeah, so it was cloned in the gene all transgenics. I'll skip this slides. Got next. Yeah, we clone a fiber entities, a vector. Go ahead. Yeah, these are transgenic plants. Okay, so you can say vector control and wild type plants, and they are two transgenic plants. They are Arabidopsis thaliana. Okay, and we saw that the plants are a little bigger than the wild type plants, and we explain a minute why. Next slide. Yeah, this is our transgenic southern blot. Now the next. Yeah, relative expression of carbonic anhydrases, the expression of gene expression increased. Next, protein expression also increased, and carbonic anhydrase activity in the plants in transgenics increased. Different transgenic lines. Next slide. Next. Yeah, so what happened? Chlorophyll content increased, okay, and amino acids content increased and protein content also increased in the transgenics. So I wondered why amino acids and protein content increased. Next slide. Next slide, we will talk about this. Yeah, also not only the photosynthesis rate not increased, but also driver the plant increased 3.6 to 4.1, and starch content also increased. Okay. 1.8 to 2.1. Next slide. We are not saying massive increase. Okay, whatever is got increase around 13 to 15%. We have tried to explain that. Okay. Okay, next slide. Yeah. So you can see here chlorophyll content, but one important thing we must say why it happened, why protein content increased. You can see malic acid, fumaric acid increased. So what happened? In phosphoryl pyruvate carboxylates, you also present also in C3 plants. Not that only C4 plants present. PEP carboxylate also present in the C3 plants. Okay. So this C3 PEP-C reacting with carbon dioxide to give you oxaloacetic acid in the cytoplasm. This oxaloacetic acid migrating from the cytoplasm, okay, to the mitochondria, okay, and reaching in going to the Krebs cycle, okay, and producing malic acid there. So in Krebs cycle. Not only that, you can therefore you can see that malic acid increased, fumaric acid increased, and aspartic acid, aspargine, glutamic acid, glutamine, alanine, all different kinds of amino acids also increased because oxaloacetic acid fed to the produced in the cytoplasm because of the bicarbonate concentration increasing could produce different kind of amino acids. Okay. All right. Therefore, photosystem 1, photosystem 2 activity also increase. Next slide. Yeah, then photosystem 2 electron transport rate increased. Yeah, if photosynthesis efficiency increase. Next slide. Yeah, next slide you can, we'll go to the end of it. Yeah. What is photosynthesis? Carbon assimilation. The wild type and transgenic plants. And next slide. And this is CO2 assimilation rate. Different carbon concentration, wild type transgenic plants. You can see how photosynthesis rate increase in transgenic plants. And not only that, we also did perform the experiment 2% oxygen, and we also saw the increase in the photosynthesis rate. From all these, we concluded that photosynthesis carbonic anhydrase could be used. Next slide. Yeah, so this is our model, final model, that carbon dioxide is a beta. This is what bidentate carbonic anhydrase we express in the cytoplasm. So CO2, the protein content increase, we produce a lot of bicarbonate, increase therefore, but CO2 plus water become bicarbonate. This bicarbonate react with the phosphonopyruvic PP by the enzyme phosphonopyruvic carboxylase to give oxaloacetic acid. Oxaloacetic acid concentration increase. That could migrate to mitochondria, where the malic acid and other organic acids increased. The amino acids increased, then protein content increased because amino acids increased. And that in turn increased the entire photosynthetic process. Yeah, I will skip this slide. The plants are also tolerant to stress. Go back to last slide. Go to last end slide. Yeah. Conclusions. Overexpression C4-5 bidentate, CA increases amino acid content, protein content, photosynthesis efficiency, plant biomass, water efficiency, and comfort tolerance to cell stress. Like C4 plants, transgenics are tolerant to salt stress. This single cell C4 approach could be replicated in crop plants, of course, to give you better product. Thank you very much for your kind attention. Yes, Arubji. Yes. Elevated carbon dioxide. Yes. So you're saying elevated carbon dioxide increasing
C3 plants for increased photosynthetic efficiency, as well as for utilizing ammonium as a nitrogen source. Okay. So, and you told sir also that when carbon dioxide concentration went down, then C3 plants they evolved to C4 plants.
Yes.
Okay, sir.
Yes.
Then, sir, why do we need to change a C3 plant to C4?
They are saying because you see, there is one enzyme called rubiscoid. So oxygen and CO2 are competing for same active site. So for the millions of years of evolution, plant didn't or couldn't change the active site of rubisco. Okay, in favor of carboxylation over oxidation. Okay, so therefore plants were forced to concentrate carbon dioxide in the vicinity of rubisco. Okay, so they formed another set of plants called C4 plants, which could fix carbon dioxide, okay, efficiently in cytoplasm, not in chloroplast, cytoplasm, transfer that carbon dioxide to the bundle cells, where the rubisco is. Therefore, CO2 concentration increased in vicinity of rubisco. So therefore, CO2 outcompeted oxygen. Okay, CO4 plant became more productive because CO2 oxygen outcompeted in CO4. So competed like carbon concentration better, CCN. The carbon concentration can increase in the therefore the plants could not change the active site of ISPO. So therefore they got other C4 plants, which even though CO it can potential for respire, but CO2 is increasing in the vicinity. So it is mimicking the high CO2 water, okay, which are there millions of years.
Think about the engineering of carbonic anhydride into the rice. So in that case, can they use the, I mean, that is the C1 tank.
See?
C1, that is submerged macrophytes.
Submerged macrophytes, yes, submerged macrophytes, you know, what do you want to say?
There is carbonic anhydride from one submerged macrophyte plants to rice.
Yeah, we can talk about it, but we don't have time for it now. Okay? We can talk about it later. Huh? We don't have time for it. Okay. Then-
Highly informative and interactive session. Moving ahead, ladies and gentlemen, please welcome Dr. Leena Tripathi from International Institute of Tropical Agriculture, Nairobi, Kenya, and she is talking on the potential of genome editing for sustainable agriculture. Please welcome her.
Sorry, I'm trying to just share my screen. Can you see my screen? Can you hear me? Hello, can you hear me? I cannot hear. Can you hear me now? I cannot hear you. Yes, Professor Cole.
Yeah, I can hear you. You go ahead.
So if you can hear me, maybe can I proceed? Maybe can you send me? Okay, let me check the chat box here. Okay. I'm not sure why I am not hearing you. Okay, as you have confirmed that you can hear me, let me proceed. First of all, I would like to thank the organizers for giving me this opportunity to talk about the potential of genome editing for sustainable agriculture. As you know that, there is a major global challenge in agriculture is the food security right now, because the population is increasing very fast, and the world population is projected to be 9.8 billion by 2050. So in order to feed those many people, we need to grow our agriculture production to be doubled. But on the same resources, because that's not going to change, because we have limited land and water and even the other resources. So our challenge remains to actually, and on top of that, there is a climate change, which is intensifying this challenge. As we already started seeing the harmful influence of the climate change, not only on the plants itself, but also on the pathogen and pest affecting the crop productivity. So what we need sustainable, in order to have a sustainable agriculture, we really need to actually close the yield gap among all the different crops, whether this is like a wheat, maize, rice, or bananas, which are mainly the staple food crop in Africa. And in order to do so, we need to actually tap on the full potential of all the tools available in our toolbox, including the new breeding tools such as gene editing. I'm not trying to say that, gene editing is like a silver bullet which can solve all the problem, but what I'm trying to say that we need to use all the tools, including the conventional technologies, and complementing them with the new breeding tools. So gene editing is actually is a quite powerful tool, and it is actually a group of techniques, not one, because gene editing includes meganucleases, zinc finger nucleases, talents, and more recently the CRISPR-Cas. And this technique is used to making like a more precise and efficient specific changes to the DNA. So what happens in this technology, that these nucleases make a double cut in the DNA strand, and then after that, the DNA, the repair machinery of the cell itself actually repairs those double-stranded break. And the repair can happen either as a non-homologous end joining. And because this type of repair is error-prone, it can create some mutations. But then these mutations are actually targeted. The second way of repair is based on the homology-directed repair, where the donor template is present. And in that case, there is normally the insertion. It can be used for the allele replacement or the insertion. So genome editing can be used to add, remove, or even alter the DNA in the genome. Okay, so genome editing is not new. Nature has been editing the genome for a very long time. And then what will happen in the past, like, when the mutations happened in the nature, and then the farmers were selecting the varieties. That's how all the domestication happened. But in 20th century, the mutations were accelerated through chemicals and the radiation. Like in 1895, there was radiation discovery. In 1927, there was first plant reported using the radiation mutation. And then in 1944, the chemical-based mutations were reported. Later on, the technologies were developed for more precise gene targeting mutation, because with the chemical and the radiation, the mutations were random. And so that's the time the zinc finger nucleases, meganucleases, and the talents were reported. But more later, like in 2013, the CRISPR technology was discovered. And finally, this technology has become the most popular genome editing approach. And CRISPR-Cas9 technology actually won the Nobel Prize in Chemistry in 2020. But that it has still not like, we will not say that's the final, because this technology is really very fast evolving. If you will see, every other week, you see something new coming up on this one. So now there is base editing, prime editing, and as well as the CRISPR activation. So new tools in the toolbox. This technology, CRISPR-Cas, has become like now the most popular technology for functional genomics as well as for crop improvement. And then there are more than 40 different plant species where this technology has been applied. You can apply the CRISPR-Cas system either for creating the mutations like deletions, insertions, replacement, which is through the non-homologous end joining, or even for the homologous directed. So in this case, this technology is based on the double-stranded cut. But later on, the CRISPR has been modified where the Cas9 is deactivated, or we call it dead Cas9. And in that case, it doesn't create the double-stranded break. And then this technology actually can be used to upregulate your gene, like a gene activation or inhibition of the gene, which is gene repressor. Another technology which has also been later reported is actually using the nickase Cas9. In that case, that there is a break only in the one strand, not in both the strands, and this is used for the base editing and the prime editing. So based on different type of editing, actually it's very important to understand what type of edits you are getting. So when there is as I explained, when there is a double-stranded break, the repair can happen two different ways. One is the non-homologous end joining, and in that case, you get the targeted but the random mutations, and most of the time these are small indels. It can be small in insertion or deletion, and we call them SDN1. So SDN, because these nucleases are the side-directed nucleases, that's SDN, and so this is type 1, so it's called SDN1. Okay, and this is mainly used for the gene silencing or the gene knockout. The second way is when you provide the donor template, and in that case, either it can be small insertion or like a copy of the repair, and this is SDN2, and then it can also be the full insertion of the gene. So this is used for the gene insertion or the allele replacement. And in this case, the donor DNA is copied. So what happens is, like in SDN1 is very similar to the mutations which somebody can obtain even naturally or using the chemical mutagenesis or irradiation, but they are targeted. So SDN1 type of mutations are not regulated as GMOs in several countries. But SDN3 type of mutation, it depends upon what is the donor DNA. If the donor DNA is from the same plant species, like for example, if I am editing banana, and if the donor DNA is from the wild type banana or another variety of banana species, in that case, this is also not regulated as GMO. But if the donor template is a foreign DNA from the different plant species, then these edited products are treated as GMOs. So quickly give you an idea what's happening globally with the legislation for the genome edited products. So you see in this map, the countries which are shaded in green, these are the countries where the guidelines are available for the genome editing. And based on those guidelines, actually genome edited crops, if there is no foreign gene integration in them, they are not regulated as GMO. The countries which are shaded in yellow, these are the countries actually where the discussion is ongoing. And then the countries which are shaded in red, these are the countries where actually the genome edited products and the GMOs are treated similar, like in Europe, South Africa, and New Zealand. So India is actually among those countries where the gene edited crops with no foreign gene integration are not regulated as GMO. And if we come to Africa, actually there are several countries like Kenya, Nigeria, Malawi, recently Ethiopia and Ghana. These are the countries who have approved the guidelines in the similar fashion. So if I give you a little bit more clearer, so like, so you see there are the conventional, that means the new varieties developed using the traditional breeding. They are not regulated as GMO. They are there is no product which are not regulated, but then there are different ways of regulation, because if a new variety is developed, each country has some way of regulating it before it gets registered and then commercialize those varieties. But they are not regulated as GMOs, okay? When it comes to the genetically modified, there is a biosafety regulation there. But when it comes to the gene editing, SDN1 type are actually not regulated as GMO in several countries. SDN1, it depends. Some of the countries, SDN1 is also not regulated, but some of the countries, they said as they only consider SDN1 as not GMO, but SDN2 are part of that one. But SDN3 are actually regulated if there is the integration of the gene is actually the foreign gene. So basically it depends when there is a DNA integration, whether that is native or foreign. Depending on that, it is regulated. Gene editing, the potentials are real. We started already seeing lot of potential, and so this technology can be used for either for the functional genomics, that is the gene discovery, or you can use it for enhancement of the nutrition, in yield improvement, making the crops resistant to the biotic stresses like bacterial disease, viral fungi, or the abiotic stresses like drought, salt, heat, cold. And there are also groups working on the herbicide tolerance. There is one very interesting note on this one is like in comparison to the GM, which were more in the big private sector's hand. Now this gene editing is actually more under the public sector, as you can see here. And then there are a public sector or the small startup companies. So that's a big difference between the two. And this technology has been applied to more than 40 different crop species in more than 25 countries and addressing these traits, which I have explained here. There are some examples we can show, like, for example, there is a high oleic soybean oil, which is the first gene edited product commercialized. This is already in the market in US, and actually this soybean has been developed using talents. There is Japan has approved its first gene edited tomato, and these are the CRISPR tomatoes, which are rich in GABA, the gamma aminobutyric acid, and it can fight actually the high blood pressure. There are also this non-browning mushroom, which has been approved in US and Canada as non-GM. And then there is rice, this bacteria blight rice, which is actually not on the market yet, but then USDA and the Colombian regulators have approved them as non-GM. Recently, there are drought-tolerant soybean, which has already also been approved in Argentina, Brazil, and Colombia as non-GM. And then in few months back, actually, there are maize, which are tolerant to maize lethal necrosis disease, has been approved as non-GM in Kenya. So I will give you a few examples from banana, what we are working on. Why banana? Actually, banana is a major staple food crop grown in more than 136 countries. And out of this, 1/3 of that banana actually is grown in Africa. India is the #1 production of banana, but in India, the banana is used more as a fruit crop. But when you come to Africa, this is actually a staple food. It's similar to the rice and wheat in India, and or the like mashed potatoes in Europe. That's how in Africa they are eat. So, you see, this is their meal. So this is all banana eat by this is like a beans, which they go. And this is another way they eat is called matoke. This is very common in Uganda. And some of the countries in Africa, like Uganda, Rwanda, these are the countries which are not only the major producer of banana, but they are also major consumption of banana. They can consume up to 0.7 kilogram daily per person, which is equivalent to 191 kilogram per year. So you can imagine how important the banana crop is. But still there is a big problem with the yield of this crop. The potential of the banana in the cooking variety of banana is 70 ton per hectare per year, but the actual is actually 5 to 30 ton per hectare. So you can see the huge gap here. The same way of the another type of banana called plantain. So it also is a huge gap. And this is basically because of the lot of diseases and pests. There are fungal diseases affecting banana like black sigatoka, fusarium wilt. There are bacterial diseases. There are also viruses like banana bunchy top, banana streak, and then there are insects, pests like nematodes and weevil. The problem is not only that there are so many pests and pathogen, but the challenge is that you will find more than one pest or pathogen actually affecting the banana crop in the same field. So that's the biggest challenge for the farmer. So there is a critical need to develop the improved banana varieties with a broad spectrum resistance to with various diseases and pests. So at IITA, we have a very strong crop improvement program for banana, and our focus is disease resistance. And here we apply all the different tools available to us, starting from the selection in the available germplasm for the resistant varieties. We also have a very big conventional breeding program for banana. Many people think that, you cannot do the breeding in banana, but that's not true. Then we also apply the biotechnological tools like transgenic approaches for using genetic modification or the gene editing. And for gene editing, we are working on several traits like banana streak virus, banana xanthomonas wilt, fusarium wilt, and black sigatoka. But for today, keeping time in mind, I think I will just give you example of 2 different traits where we have more advancement. So we have established the CRISPR-Cas9 based gene editing for both banana and plantain. Let me explain here. Banana is triploid. Most of the cultivated bananas are triploid. Actually, for the breeding, we use the diploids, which are the banana progenitor. So when in the triploid, actually, they have 2 different types of genome, A genome and the B genome. And so the most of the dessert type of bananas, like which we use as fruits, those are the triple A's. The cooking varieties are mainly the AAB or ABBs, and the plantains are AAB. So we established the genome editing system for all different groups of bananas with different genomic groups. Okay, and so we use like PDS, which is a phytoene desaturase, which is involved in the beta carotene pathway, and as a visual marker. So if the PDS, that is phytoene desaturase, if the PDS is functional, plants are green. Okay, if we knock out the PDS, then we make the albino plants. So it also depends because there are 3 alleles. If you knock out all the 3 alleles, then you get this type of complete albino plants. If few alleles has been knocked out and some remain, then we get these type of chimeric plants. And this is the wild type where we have not knocked out the PDS. And then we have actually, after that, we have done the target sequencing. And this, based on this sequencing, we identified the targeted mutations. So once we established the system, we applied that system for making the bananas resistant to one of this virus called banana streak virus. So banana streak virus is a double-stranded DNA BADNA virus. And this virus has a problem that it integrates into the B genome of banana. So this is a problem only in the banana varieties which has B genome. So banana streak virus it has a monopeptide genome, and basically it has 3 open reading frames. Open reading frame 1 and 2, these are small open reading frames, and they are mainly involved in the virion assembly. Okay, open reading frame 3, which you see here in red, this is a big open reading frame. And basically it is a polyprotein which cleaves later on at the aspartic protease to make this all the functional protein. And this is the open reading frame where all the essential genes are there. Like for example, the movement protein, the coat protein, aspartic protease, reverse transcriptase, and RNase H. So this virus, when it integrates in the B genome of the banana, it first of all, it integrates as a specific locus in the chromosome 1. And then it always integrates in a multiple copies as a direct and the indirect tandem repeats. When it is integrated, the banana, it sits very quietly. It has become like an endogenous part of the banana genome, and banana doesn't show any symptom when it is integrated. But what happens when the plant is stressed, and these stressed can be the environmental stress like increase in temperature or drought, or the stress can be the hybridization during the crossing, or the tissue culture. This is a vegetatively propagated crop. So you use tissue culture to multiplication. That's time when plant treat that as a stress as well. And during the stress, this integrated virus becomes activated as in the episomal form. And then the plant starts showing the symptoms like this yellow streak. And then if it affects the yield. So because of this activation, and then at some phase not showing symptom, another phase showing symptom, it has become a very challenging for the breeders in the breeding program, but also in the dissemination of the hybrids. Many regulators in several countries actually doesn't allow the movement of these plantain, particularly plantain hybrids, from one country to another because of this challenge of the virus. And then another thing is that, Musa balbisiana, which is a progenitor, it has BB genome. This is a very good parent because it has resistance to most of the diseases, but breeders cannot use this one into their breeding program because of the issue of this banana streak virus there. So that's why when we have established the genome editing, we felt like, okay, let's target this one. So the approach I am using here is like, we are trying to knock out this viral sequences so that it doesn't activate into the virulent viral, the infectious viral particles. So the approach I'm using it, I'm like, knocking out all the 3 open reading frame. And so we have designed the guide RNA from open reading frame 1, 2, and in open reading frame 3, actually, I picked aspartic protease because my intention was that if I can knock out aspartic protease, which is required to make this polyprotein into the functional proteins, then I can actually make all the proteins non-functional because there will be no cleavage at the aspartic protease to make the functional protein. So we worked with a variety called Gonja Manjaya, which is a plantain very commonly grown in East and Central Africa. And then first what we did was we have to characterize this plant to see that what is the integration pattern of this virus. So we did a lot of molecular characterization to see this type of integration pattern, which actually what we are thinking. And then we designed the 3 guide RNA, we multiplexed the guide RNA, and we delivered the guide RNA and the Cas, which we call now CRISPR-Cas reagents, into the cell suspension of banana. So these are like, single cells of banana in the liquid form, and then each cell has the potential to grow into the complete plantlets. So we have delivered these CRISPR-Cas reagents into this and then regenerated the plantlets. After that, we did quite a lot of molecular characterization to find out, to detect the mutation. So we did later on, we did the sequencing. We didn't do the full genome sequence. What we did was the target sequencing from the targets where we were expecting the mutation. So our first target was like a open reading frame 1, and the second is the open reading frame 2. Because these targets were very close, I was expecting a deletion of 198 base pair of the viral genome from there. And that's what I was expecting. Actually, I saw the same thing in our mutants. And in the open reading frame 3, because there was only one guide RNA designed there, so we were expecting small indels, and that's what we saw. So after that, we actually selected the mutants, and then, we have done a glass house evaluation. And here we mimicked the drought. So, we didn't water these plants. And so because drought is a stress, so this is our wild type, which actually really showed the very nice yellow streaks, like the symptom of the virus. But then several of the mutants actually didn't show any symptom after the stress. And we have further confirmed that as like virus load, and we didn't see any virus in these mutants. But the 2 mutant actually showed some mild symptoms. So I was very curious to know what happened to these mutants, why they were showing the mild symptoms. So after characterization, we noticed that these 3 mutants, which had the asymptomatic mutants, which had no symptoms at all, actually they have the mutation in all the 3 open reading frames. Whereas in these 2, there was a mutation only in open reading frame 1 and 2, but not in the open reading frame 3. But you remember I mentioned that open reading frame 3 plays a crucial role because that's where all the functional genes are there. So we have now the proof of concept that, if we can create the mutation in the aspartic, we make the non-functional aspartic protease, then actually we can disrupt the function of protease and we can inactivate the virus. So this is now we are working with this all the plantains to take them to the field to confirm our theory there. The second story is from the banana Xanthomonas wilt. This is a very serious problem because it is devastating banana plantation in the Great Lakes region of the East Africa, which is also the major banana producing area. The losses can be 40 to 80% in total in depending upon the country. But when you go to the field, actually is an absolute loss because once the disease is there, it spreads very fast and it wipes off the whole field of banana plantation within few months' time. And all the cultivated varieties are actually susceptible to this disease. There is only wild type banana, Musa balbisiana, which has found to be the resistance. There are few other wild type bananas which are tolerant to this disease. And the overall economic losses are estimated to be 2 to 8 billion over a decade, which is a big amount of loss for African countries. And as I mentioned, all the banana varieties are susceptible. And basically, you see this type of scenario because once the field is infected with this disease, there is no other option than to actually uproot the plants and leave the field fallow for a minimum of 6 months before it can be replanted by another bananas. So what we did was we have actually studied the Musa balbisiana at the molecular level to understand what makes this wild type banana as resistant to this disease. So we did the comparative transcriptomic by infecting these Musa balbisiana with the bacterial pathogen and compared that with the susceptible variety. And we did that at the early stages. And based on this comparative transcriptomics, actually, we have identified a lot of targets which we can use it in our gene editing program. Some of them are the susceptibility genes, which are actually involved in the development of the disease. Some of them are the transporter genes or the transcription factor involved as the negative regulator in the defense pathway. And right now we are testing about 10 different susceptibility genes by knocking out one by one and test. So what we are trying to do right now is like, this is the wild type. We have the full information, what is making these plants resistant, and these are our susceptible cultivated varieties. So we are transferring the information, not the DNA or anything, just the information from this one to here based on the gene editing. So either we are overexpressing the defense genes, which are the endogenous genes, but we are upregulating them using CRISPR activation, or we are knocking down the susceptibility gene using the CRISPR Cas9 system. So one example I will show, which is the DMR6. You know, DMR6 is a downy mildew resistant 6, is a susceptibility gene, is a very well characterized susceptibility gene in Arabidopsis and rice. And this susceptibility gene is actually activated during the host pathogen interaction. So it normally overexpressed during the pathogen infection and then suppress the plant immunity. So what we did was we find out the ortholog of this DMR6 in banana, and we actually created the phylogenetic tree using like the mapping it with the Arabidopsis, with the different Musa acuminata, balbisiana, the nicotiana, and as well as tomato. So if you want to know more detail about this, it is published in Plant Biotech Journal. So you can also see. So based on this phylogeny, actually, we picked ortholog. So we found 7 different orthologs of DMR6, and we picked one specific ortholog and then tested it by qRT-PCR. And then we saw that, yes, our susceptible variety, when it is infected by pathogen, actually this gene goes up by 60-fold, whereas this Musa balbisiana, which we were using as control, actually has no differential expression. So this has given us the confidence that, you know, if we knock out this Musa DMR6 ortholog, then maybe we can make the plant resistant to the disease. So that's what we did. We designed the 2 guides targeting the DMR6 ortholog, and we created the DMR6 mutants. We did the sequencing to detect the targeted mutations, and so after that we tested these plants using a rapid bioassay in the tissue culture. So these are our control wild type plants, where you can see they have completely wilted after challenging them with the bacterial pathogen, and these are our mutants which were showing the resistance. So after further, what we did, we planted these ones in the greenhouse and challenged them with the bacterial by like, you know, artificially infecting them with the bacterial pathogen in the leaf petiole. And these are our control wild type, and they completely wilted. They started showing symptoms in 10 days post inoculation, and within 1 month time they were completely wilted. But these plants, we actually grow them up to 90 days and they didn't show any symptom at all. So we also wanted to see that whether knocking out this DMR6 will have no negative impact on plant growth, because, you know, most of the genes, they are not only involved with one function, they are involved with several functions as well. So, but under greenhouse condition, actually, we didn't see any detrimental effect of knocking out this DMR6. And the main reason about that is because, you remember I said that there are 7 orthologs, but intentionally we didn't knock out all the 7. I only knocked out 1. So that, you know, it doesn't have impact on the plant growth. So now we are ready to test them under the field condition to check like further the stability of the trait and also the effect on the yield and So one thing which I would also like to say is that the mutations created through the CRISPR-Cas are actually quite stable, which we have already tested. We have multiplied these our mutants for the 7 different generation and then tested, and we find them they have the same mutations even after seven generations. So this is my last slide where I would like to mention that, because this is a very new technology, so in order that farmers to adopt not only farmers but the consumers to also start farmers to grow and consumers to start eating these type of varieties, you need to do quite a lot of public awareness and also need to create the capacity. So we are very heavily involved in that one. We do every year 1 or 2 workshops where we invite the regulators, the journalists, the public, like a farmer association representatives, and we provide them more information on this technology. We are also building critical mass in Africa. So, we are training a lot of students and the young researchers in our lab. And then recently, actually, we started a program, which is an African plant breeding association, where actually we are collaborating with UC Davis and UC Berkeley. And you know that UC Berkeley, this is the origin of the CRISPR technology, because that's where the Jennifer Doudna is based. And then we are right now, this is a 5 years program, and every year we are training 20 young researchers. These are scientists, they have PhDs already, and they are working with the national system in African institution. And we will be training 20 scientists for 5 years. So in total, we will be training 100 scientists in our lab there. And so the first batch of the scientists are actually under training currently in my lab. It started from the 23rd of January. So the key take-home messages are banana production is really constrained by several diseases and pests. CRISPR-Cas technology has rapidly become the most popular genome engineering approach because it is very simple, is efficient, is easy to adapt, and is very specific, and you can do multiplexing. So right now we were multiplexing the different guide RNAs for one trait, but our intention is that we will multiplex is to the different pathogens as well. And so, in banana, there is a need to develop the varieties with the multiple and the durable resistance to combat the several biotic stresses. And that's where the CRISPR plays a major role. We have already demonstrated that the CRISPR-Cas based genome editing, we can inactivate the banana streak virus from the genome, which was integrated there. We have also demonstrated that the DMR6 mutants of banana showed enhanced resistance to banana Xanthomonas wilt. And genome edited crops, we are expecting our bananas as well, with no foreign gene integration, are not regulated as GMO in several countries. So in the end, I would like to acknowledge my team, my plant transformation team, the bioinformatics team, and also the partners from UC Davis and the Alliance for Science, and the financial support from USAID and the CGIAR research program. Thank you very much. I will be more than happy to take any question if my microphone works well. Thank you very much for your attention. I cannot hear you, but if you can write, if there is any question, please write in the chat box and then I can respond.
And one thing which I would like to add to the students or those who would like to work on, like two things are very important if you want to work on genome editing. One is that there should not be even novel combination of genetic elements from different organisms, as it happens in case of GM plants. And as a result of that, only the GM plants are regulated. Has anybody thought of it that why do we regulate after all GM plants? Because there is a novel recombination of genetic elements taking place from different organisms, which otherwise may not be possible for millions of years to occur in nature. But since we're doing it, so therefore, but here in case of SDN1 and SDN2, there's no such novel combination of genetic elements taking place, that we are not taking organisms, different genetic elements from different organisms. We are not making a recombinant DNA in the final product. I'm talking about it. And then we are not therefore calling it a GMO. And therefore in many of the countries, as Dr. Lina Tripathi mentioned, these SDN1, SDN2 categories are considered as non-GMOs. And the second thing is that this is very important for the students if they may like to think about later working, that why do we have no regulation in case of SDN1 and SDN2 in genome edited plants? The question was asked by one of the participants here from Dr. Kole, whether we should worry about antibiotic resistance marker gene, whether it is automatically after certain selfing, it is, kind of, removed, from within the plant. We do use, actually speaking, antibiotic marker genes, which are required, as we normally do in case of developing transgenic plants. The first half as shown here is we do develop a transgenic plant initially, when we do genome editing. But later when you self it, that whole reagents, used for like antibiotic marker gene or all the CRISPR reagents, they are removed. In fact, they are segregated out after do selfing. And that's what is shown here. So you can see here, the genome edited plant with transgenes is a transgenic plant. It's a transgenic plant. When you self it, all those antibiotic marker genes or all the CRISPR reagents, then they are segregated out when you do the selfing. And therefore you select only the plants as shown here in red, the genome edited plants without any transgene. So it has only the edited portion, as I said before, SDN1 or SDN2. I'm talking about it here. And this is what is shown here in this particular slide. And this is the advantage of genome editing. And as a result of that, you can see here, this has a great relevance. Actually speaking, when you talk of, use of genome editing for climate change, if you want to enhance water use, water conservation in rice, it has been demonstrated. Engineering microbes to curtail dependence on synthetic fertilizers. I mentioned before, that's one of the opportunities. Yes, it's been done. And also, can we have crops fixing more CO2 more efficiently? And this is what actually Jennifer Doudna, one of the, lady scientist who shared the Nobel Prize, in chemistry in 2022, she also feels, that it has a big potential for use in climate change adaptation. And then in India, actually speaking, there's a lot of work going on already in about 25, 26 crops. The genome editing work is going on in India. And here, this is our Honorable Prime Minister making a statement in the parliament saying that, yes, the farmers should get the first drought resistant and genome edited rice variety by 2026. And this is being developed, actually speaking, by Indian Agricultural Research Institute, by Dr. Viswanathan's group. This is, showing an advantage of increased water use efficiency by at least 25% and saving, therefore, a lot of water. And not only that we try to increase the use efficiency of water, there is also a possibility we can try to, increase the efficiency of nitrogen or fertilizers or potash utilization. And on the right panel, you can see here, as Dr. Robert Henry also mentioned, we can convert many of those wild species into cultivated rice, which will only take about 2 to 4 years instead of thousands of years, which, normally domestication will take place, that much in time. Here, we can try to do it in 2 to 4 years. And when you talk of information available or the genetic material available, we are very rich in India. We are the second largest gene bank in the whole world. We have compiled the whole information of wild relatives with all the characteristics given here, of the cultivated plants in India. And germplasm to genome engineering is the final message, which I think is very important, because natural genetic variation which is existing in the germplasm, we can now try to mimic it through genome editing into our new high-yielding genotypes. And this is what actually the message I'm trying to give here through this particular slide. But finally, we are so very happy again that India has exempted SDN1 and SDN2 category of plants, from biosafety assessment. You don't have to go through biosafety assessment. If I may ask now any one of the students why we don't have to go through the biosafety assessment of SDN1 and SDN2. And therefore, India has exempted these plants from this regulation, which is otherwise applicable to GMO. Simply because, there is no novel genetic combination taking place here. And in the final product, there's no such foreign or marker gene or nothing there except the native sequences within the particular crop species have been edited or deleted or corrected the gene function. So this is what actually I'm showing you here, all the details, that Government of India has permitted, excuse me, next slide, please. And now, so this is the slide I'm showing here. In case of GM crops, the route was from IBSC to RCGM to GAC. This was the route. GAC is the final authority to approve the GM crops commercialization, as they have done recently. I give you an example of GM mustard. But in case of genome editing, IBSC to play a key role, only IBSC. RCGM and GAC are not coming into the picture to take a final decision when we talk of release of these genome editing plants in India. And this is exemption under Rule 20 of the Rules 1989. And therefore, there is no need. And finally, important is, therefore, once we have got this kind of an advantage, as Dr. Leena Tripathi also mentioned, on the world map, India is green. Once we have that advantage, then what are the crops and the traits for which we should be working on in India? And these are the traits in a discussion with several of Government of India scientists and even secretary level. This was the discussion, and we identified these are the crops. I know there's a lot of work going on rice. There's a lot of work going on other crops. But important is what for India? Mustard, soybean, groundnut as oilseed crops, pulses, chickpea, pigeon pea, and green gram. And of course, also we cannot forget rice, wheat, some of the cash crops and even horticultural crops, which are very important. But finally, for the sake of the researchers in our country, we have come up with a roadmap, actually speaking in this kind of an article in Current Science, what is that what are those crops and which are those priority traits and what are those genes and whether SDN1 and SDN2, whether genome editing in the exon region or genome editing in the promoter region, where it has to do what is all given here in this particular slide, in article. I'll be happy to share with you all that article. So this was all in brief, and I thank you, friends, very much. And again, thank organizers for giving this opportunity. Thank you. If there are any questions, if I have time, I believe, yes, I think I finished on time. And if there are questions, students, yes, please.
Thank you for very insightful presentation. So my question is, group of scientists who are working. So is the genome editing is dependent on that?
Not really, not at all.
So how do you select which genes are important?
No, functional genomics, true functional genomics. While even those markers and QTLs which have been identified, which are being used by the molecular breeders, you're right, which is going on. But if you do fine mapping of those genes, and finally you come down to the level of a particular gene, you validate its function, then using transgenic approach in a model system, and then once you identify a particular gene and a particular genetic element within a gene, it's possible, then of course you can apply even genome editing. But otherwise, important is in our country, we should give an equal emphasis on even identifying a particular genomic region or the gene, which is important and associated with a particular trait before we do genome editing. Like many of these disease-resistant genes, they have already been identified. Like Dr. Vishnathan, I give an example of rice. He has identified one particular gene, which is a negative regulator, of drought tolerance. So if you knock out its function, the plants become resistant. So that way, yes, it is important to identify genes. And therefore, after genomics, that's what I was talking to my friend, Dr. Vaishnav Tripathi, sitting right here, that functional genomics is something which students should really come forward. And using the genetic material, which is available with us, genetic diversity in our gene banks, that should be utilized to as extent as possible. My pleasure. Yes, please. Yeah, after him. Yes, please.
In the committee, health ministry or health department for release of mustard,
Yes.
Yes,
Yes.
Do you have the involvement of high technology research institute and another?
You know, actually, you know what? There is a system. I said before, there is an Environmental Protection Act, Government of India, 1986. Under that act, we have got rules. We call them as rules 1989, right? And those rules, under those rules, we have guidelines. Under those guidelines, we have got standard operating procedures. And also under that act, there are 3 different committees: Institute Biosafety Committee, Review Committee on Genetic Manipulation, Genetic Engineering Appraisal Committee. And all these committees have experts from diverse fields which you're talking about.
Specifically, High Technology Institute at Anantapur and AP has been consulted regarding the composition of mustard.
Right. Yeah, all those tests are done. For example, composition analysis were done by National Institute of Nutrition.
Nutrition.
Yes, in Hyderabad. Yes.
Yeah. Oh, nutrition, I'm asking for the higher technology.
See, you don't have to have a kind
It's
of,
also a national institute and
wherever there is a requirement, only that National Institute will come into picture. If there's no requirement, why should it come into picture? If something is required for a particular crop, for a particular trait, involvement of that particular institute, yes, it is very much important. We should have it. Otherwise, why? Otherwise, not.
to answer the public opinion.
Yes.
Public opinion is not totally for gene modified crops. So that's why it may be necessary to involve all technology institute also, my opinion.
Every institution can be involved, and if they are invited to give their own comments. So some of these, in fact, documents are always available on the website of the Ministry of Environment and Forest and Climate Change. One can always visit and give the comments.
Okay, thank you.
Yes, welcome. Yes, please.
Thank you for brief presentation. Because from morning we are focusing on the climate change. I have one question actually. Does resistant breeding or maybe transgenics really work in altered climatic conditions? Because there may be elevated CO2 levels and a slight increase and decrease in the temperature. Does the resistant breeding really works in that altered climatic scenario?
You know, actually speaking, what we are trying to do is, whether conventional breeding or any other method of molecular breeding people are trying to use, they are doing resistance breeding, yes, very much. And when we're talking of testing them, they are being tested under different environmental conditions, which are prevailing today. We are not going to test them for the futuristic point of view. I can understand what you're trying to say, that are we testing them, for example, if you're predicting that temperature is going to be higher beyond this. That's not actually speaking being done at the moment, but under the environmental conditions which are there, under different agroecological conditions, they're all being tested there. We have in our Indian Council of Agricultural Research and Agri trials, we call them, which are scattered depending upon the crop, different agroecological regions. And all those crops, before they were bred and varieties released, they're all tested under those conditions. Whether you talk of resistance breeding, you talk of advancement in yield, or any other trait for that matter. So there's a wonderful mechanism we do have. But I take your message, yes, probably I will check whether are we going to initiate something of that kind, you know, under stimulated environmental conditions, which we predict, let us say, going to happen, temperature or CO2 from now 10 years after.
Yes, sir. There is one report actually in Denmark. One weed is there, one legume is there, Stellanthus cabra. Initially, it is having a major disease that is anthracnose, Coltotrichum bleosporides. Slightly rise in 80 ppm of that CO2 levels. The symptoms are more seeing on that resistant breeded varieties rather than susceptible varieties.
I'll be happy to have that paper from you as a reference if you can pass it on to me.
Okay, sir.
Sure.
I will pass it. Okay.
Thank you. Okay. If there are no more questions, once again, I thank the organizers for this opportunity, and I'm happy to see 3 students still left behind. Others have left because it was lunchtime. They were feeling hungry. So my special thanks to these 3 beautiful students still, bearing with the presentation. Thank you very much. If you have any question, comment, you can still make it later during the lunchtime. Thank you all. Thank you very much.
Thank you so very much, sir, for the resounding piece of presentation. As sir will leave early for his other commitments and the schedule for the day. To felicitate sir, may I welcome on the stage Dr. Arup Kumar Mukherjee, Honorable Principal Scientist, ICAR, National Rice Research Institute, Cuttack, and Honorable President of Society for Agricultural Research and Management. I request sir to kindly join us on the stage, and Professor C. Kole, along with Engineer Dibyanshu Dash, to kindly come up on the stage to felicitate Professor K.C. Bansal. I request sir to kindly be on stage. As he is leaving early due to his other commitments, it's a small token of appreciation from the side of Agri Vision 2023. Yes. We are honored to have you here, sir. This is the Lifetime Achievement Award. I would request all of you to kindly give a standing ovation for Professor K.C. Bansal. We congratulate you, sir. A continuous round of applause for Professor K.C. Bansal, everyone. Thank you so much, sir. Thank you, Dr. Mukherjee, Professor Kole, and Dr. Das for doing the honor. Next up, we have Dr. J.L.N. Sastry, former CEO, NMPB and Managing Partner, Jatas Ayurvedic Healthcare Systems. Let's welcome him. He is joining us shortly. He will be joining post lunch. I would request all of you to kindly have your lunch. Thank you so much. Let us gather
Can you give me an opportunity to have presentation today
Sir, may I know your name?
in the absence, please? In the place of absentees.
Yes, may I know your name, please, sir?
My name is Subbarao, not listed here.
Okay, okay, I'll
That's
talk.
why,
Okay, sir, I will talk and I'll sit in.
yeah, you talk to Dr. Sardas and make an.
Yes, yes, yes. You can.
I'm Subbarao.
Yes, sir, can you please send your topic to him?
He-
Can you please send your topic to him so that
I sent a copy yesterday night.
only the name, actually you had sent, okay, okay.
Huh?
You had sent him.
I've handed over to him.
Okay.
And I have another copy. If you want, I will give you.
I only want to know the name of your topic.
Okay, my name is M. Subbarao,
Yes, sir.
retired Deputy Director Agriculture, AP.
Deputy Director,
Presentation on agriculture and rural development.
Agriculture and?
Rural development. The proper title.
Mhm.
It is the proper title.
Okay.
All the presentations are going.
Retired Deputy Director, Agriculture, which department?
Yeah.
Which department?
Agri department.
Odisha.
No, Andhra Pradesh, AP.
Government of
Andhra Pradesh.
Government of AP.
Yeah. I mean, retired people, not because connected to either government or department.
Okay, okay, sir. Please have a seat.
Oh.
Check.
Basically laptop I use for this presentation program.
Okay.
So they just have to do it over here and
This is the copy.
You have it. Okay, okay. You have it with you. I have your topic
Okay.
already.
And also, somebody was advising, he will include in the soft copy
Okay.
of the presentation.
Okay.
Printed copy printing complete हो गया, इसलिए soft copy include कर सकते हैं.
Okay, okay. Ladies and gentlemen, welcome back after the lunch break. I hope we were able to build some network during the networking lunch, and I cannot emphasize how important it is to have a network when it comes to agri business, especially. Moving ahead, next up we have Dr. Ganesh Babu, Standard Biotools, Singapore, and he is talking on high throughput quantitative polymerase chain reaction for plant and animal genomic applications. Give a big hand for Dr. Ganesh Babu, everyone.
Yeah, thank you, everyone. So my name is Ganesh. I am from Standard Biotools. If you haven't heard the name Standard Biotools before, you might have heard the name Fluidam actually. That is our company. So we have our offices in San Francisco and Canada and then Singapore actually. So we work broadly in two areas. One is high throughput genomics and another area is proteomics. So in today's my talk, I will be talking about how we use various tools in high throughput genomics. It's not moving. Next slide. So in today's presentations, I will be talking about high throughput qPCR platforms for a plant and animal genomics applications. I hope you might have used a PCR or real-time PCR, but it's not just yet another qPCR machine. So it's entirely very unique and very innovative tool for plant and animal genomics. So when I talk about in a detail, you will know why is it innovative, how it helps for researchers in plant and animal genomics application. Next slide. So in today's presentations, I will be talking about what is unique about Standard BioTools qPCR system, and then how it works. Basically, I will be explaining about the technology that begins the system. Then I talk about our system X9 real-time PCR system. Then finally, I will be showing some of the applications that commonly used in plant and animal genomics research. Finally, I will be taking up the questions. Yeah, please, next slide. So why is Standard BioTools? There are many companies that makes qPCR around the world. So why? What is unique? And what is the value proposition that the Standard BioTools brings to the world? So next slide. So if you have used a real-time PCR, you know one of the shortcoming of the conventional real-time PCR is the number of target you can study. So let's say you wanted to do some real-time PCR experiment. So if you use a conventional qPCR system, so you can go maximum of 6 target. So you can study only maximum of 6 target if you use a conventional qPCR. Why? Because most of the conventional qPCR system has only 6 fluorescent channel. So the moment you wanted to study more than 6 target, you may not able to use a conventional qPCR. Let's say if you wanted to study more than 6 target from any given sample, you may not able to use a conventional qPCR, or if you use a conventional qPCR, you need to run a multiple reaction. This is one of the shortcoming of conventional qPCR because of the limited fluorescent channel that is available with the conventional qPCR system. And even though there are a 6 fluorescent channel, you can multiplex up to 6, but it is not also easy, because when you do the multiplex reactions, you need to optimize the conditions for the multiplex qPCR. That is also a challenge. So let's assume you have a sample and you wanted to study 96 target from conventional, you wanted to study a 96 different genes or 96 different target. If you use a conventional qPCR system, let's assume you are running your experiment in a 96 well plate. So that means that only you can run one sample. You can run only one sample if you use a conventional qPCR when you want to study 96 target. So for multiple target, you need to run a multiple plate. That is one of that limits the number of sample you can run in your experiment. Next slide. So next slide. Next slide. So we solve one of these unique problem. Actually, you can study maximum of 96 or 192 targets simultaneously. And even that is in a single plex reaction. You're not doing multiplexing. So we solve 2 problems. So the first problem we solve, you can study up to 192 target simultaneously. And another one problem, you don't need to worry about multiplexing, because all the reactions happens on a single flex. So these slides is nicely summarized about the power of Standard BioTools technology. So this is not the created picture. So this picture is sent to us by one of our customers in US. So in this picture, you can see there are 384 well plate. So these plates were used by one of our customer in US to do genotyping. So they need to generate 0.5 million genotyping data point. They need to generate a 0.5 million genotyping data point. In order to generate, they need to run 384 well plates. So that means that they need to run conventional qPCR, real-time PCR 1300 time actually. So if they wanted to run this kind of a project, they need to run 1300 time actually. Let's assume as a human, even if you run 3 plates per day, how many days these kind of a projects is going to take actually. So Standard BioTools solve one of these unique problem. Next slide. So whatever you did with the 1300 plate, that can be done with as low as a 54 run actually. So whatever you did with a 1300 run, that can be scaled down to 54 run. That's the power of the technology that's begun. So combinations with our convention, our high throughput qPCR system X9 with our technology. So whatever they did with the 1300 run, that can be scaled down to 54 run. How is it feasible? So what is behind the technology? So when you understand the technology, you know how it works, how we can scale down. Next slides. Yeah, next slide. So our technology is based on microfluidics. So microfluidics is nothing but you miniaturize everything. So whatever you do, you miniaturize everything. But it is not just microfluidics. So the company has invented and innovated in a very clever way that you can simultaneously study multiple targets. So our technology is based on integrated fluid circuit. So our technology is based on integrated fluid circuit. So when you do the real-time PCR, you use 96 well plate or a 384 well plate. But in this one, so instead of using a 96 or 384 well plate, we use a special chip we call as a integrated fluid circuit. In a short form, we call IFC. So in this picture, you can see the middle black color portion. That's called the IFC. So that is a very tiny 3 centimeter into 3 centimeter square. That tiny portion in the middle, that is only 3 centimeter into 3 centimeter square, where there are more than 9,000 PCR reaction chambers. In those tiny space has more than 9,000 PCR reaction chambers. So that means that within that tiny space, there are going to be more than 9,000 PCR reaction. On the other, if you look at on both the sides, left hand side and right hand side, you can see there are an inlet. Those inlets are called a assay and sample inlets. So there are in this particular represented picture here, there are a 96 assay inlet and 96 sample inlets. So you can load simultaneously 96 sample and 96 target. So each target will be integrated for each sample. So that means that it allows you to create 96 into 96 reaction combination. Go to the next slide, please. Yeah. So here you can simple comparisons on the left hand side. If you use a conventional qPCR, so you are, let's say, assume you wanted to study 96 target, you can run only one sample. But if you look at here, so in the standard BioTools solutions, so you can study 96 sample against 96 target. One more plus. So in a simple way to say, you are running only 96 reactions actually. If you use a conventional qPCR, you are running only 96 reactions or 96 data point. But when you run with the standard BioTools qPCR, so you are going to get about 9,216 data point or reactions. Go to next slide. So in a nutshell, so these high throughput systems allow you to study multiple data point and multiple target simultaneously. So it is scalable while it's providing the high throughput option for you. Go to next slide. Next slide. So to say it in a simple way, so one run of IFC, that is equivalent to 96 well plate. So that means that you need to run 96 times conventional qPCR to get the same amount of data you get from X9 system, our system. So I repeat again. So if you want to get the data output you get from one run of X9, that is equivalent to 96 well run actually. So that is what this picture represents here. Yeah, next slide, please. And again, it is not only throughput, it's also allow you a cost saving. It's not only throughput, it's allow you to cost saving. So I'm going to talk about and showing in a quantitative way how it allow you to do the cost saving. So let's assume you have 1,000 sample. You wanted to target 96 target. It can be gene expression or it can be genotyping target. So that means that you need to generate 96,000 data point. So you need to get a 96,000 data point. If you use a conventional real-time PCR and run with a 384 well plate, it's going to take 250 run. So if you use a conventional qPCR, it's going to take 250 run to complete this kind of a project. And for the qPCR master mix alone, you will end up using almost 1 liter of master mix. For this kind of a project, you will end up using 1 liter of master mix. If you look at the master mix price alone, the list price alone, it's going to cost 82,000 euro. So for this kind of a project, the master mix alone going to cost 82,000 euro. And it's not only the money and amount of reagent, the time also to complete this kind of a projects with the conventional qPCR, that's going to take almost 5 months of your time to complete these projects. So far you have seen how much time, how many run, and how much amount of master mix, and how much amount of money you need to spend to get this much data. So one more slide, please. Yeah. So here it's a comparison. So let's assume if you run that with our standard BioTools qPCR systems, same amount of sample, same number of targets, and it's going to take only 11 run, 250 run versus 11 run, and amount of master mix. Because the reactions happens on a microfluidics, it's going to consume very, very nanoliter amount of volume of the master mix. So that's what it's going to consume, only 2.64 ml, less than 5 ml actually. So 1 liter versus less than 3 ml of master mix for this kind of a project. So what you are spending for master mix going to be drastically scaled down to $430 actually. So the 30 euros actually. So compare the price of the master mix you will end up paying for this kind of a project. And more importantly, time to data. So it's going to take only 4 days to complete these kind of a projects when you run with this system. So that's the power of our system. Next slide, please. So this slide, there will be a video, just play that. So you can you just bring your cursor over the thing you can see on the bottom, and you can click, then it will play. Yeah. Okay, go to the next slide. Yeah. So here I'm showing our X9 instruments. Yeah, next slide. So this is our PCR system. So this is a system that we use to run these IFCs. So this is called a X9. It's a real-time PCR system. So it can be used for your quantitative and qualitative experiment. So you can use this system. Let's assume you are running a gene expression. It can be used actually for your gene expression experiment because it's a real-time PCR system. It measures the expression in real time. And it's not only for the gene expression, it can be also used for genotyping. So let's assume if you are doing any genotyping, it can be used for your genotyping experiment as well. And so if you have this kind of a system, in a 1 day, you can generate more than 46,000 data point. In a 1 day, in a 8-hour shift, you can generate more than 46,000 data point. And you can simultaneously study 96 target. And you don't need to do any optimizations. And again, this system is easy to operate. Everything can be operated from your touch screen. You don't need to do even more. System is more intelligent, and that also recognized based on the IFC we use, what kind of experiment you are running, and that will select automatically the protocol that need to be selected for the experiment. So go to the next slide, please. Yeah. So here is the workflow, how the systems work. This picture shows the complete workflow of the system. So you can, first thing, it started from extracted DNA. So the DNA can be extracted from your sample of interest. It can be a plant, or it can be a animal tissue or human sample. So extract the DNA. So DNA is a starting material. So the next one is designing the assays. So you can either you can design your own primer, or you can use a TACMAN assays, or you can also use we have our own genotyping solutions that's called a SNP type assays. So we have an online tool where you can design a primer for your experiments. So that tool is called a D3 online tool where you can design assays for your experiment. So once you have assays and samples are ready, you use a multi-channel pipette and transfer the sample to IFC. So you can use a multi-channel pipette to transfer the samples and assays. And once you transfer the sample, you put it into the qPCR system that's called a X9 system. And you then in 2 hours, 15 minutes time, so you will have a data point of more than 9,000 reaction data point in a 2 hours times. Then finally you can analyze the data using our online tool that we have online software that can be used for analyze. So we have 3 different types of IFC to meet different throughput. The first, so if you look at the bottom, you can see that there are different numbers that represents the different types of IFC. So for an example, 96.96 means you can run 96 sample against 96 target. 192.24 means you can run 192 sample against 24 target. And then 48.48 means that you can run 48 sample against 48 different assays or different primers. So here the first number that denotes the number of sample you can run. The second number denotes the number of assays you can run actually. Yeah, go to the next slide. So here I'm going to show you some, so there are more than thousands of publications that have used our platform around the world. It's not a new technology. It's been in the market for 10 years, and worldwide, many researchers have used and published thousands of papers. So here I'm going to show some of the relevant example that is related to plant genomics applications. Go to the next slide, please. Yeah. So as we know that when it comes to plant, so the plant biologist used various techniques for molecular biology techniques in a different stage of their applications. So molecular techniques are used in plant breeding and pre-breeding stages. It is used during the breeding, and it is used during the quality control stages. So in different stages, different tools are used. For an example, SNP genotyping by PCR, it's commonly used during the breeding stage, and then high density markers like a whole genome sequencings or genome-wide microarray genome-wide association studies are usually done during the pre-breeding stages. So during the pre-breeding stages, we need to study lot of markers because we need to screen the plant of our interest for our experiments. So you're screening the stock that's required for your experiment. But when it comes to breeding, you don't need to do whole genome sequencing or microarray. First thing, it is costly. But then during the breeding stage, you are going to screen hundreds of plants to select your marker of interest for the marker resisted selection. So you need to screen only limited marker. So those times you can't use a conventional PCR, you need a more marker. So that stage, standard BioTools qPCR system X9 is commonly used during the breeding stage. Apart from breeding, it is also used during the quality control stages. So let's say you are making a new seed, you are about to release the seed. So if it is a GMO or it is a newly developed variety, you need to test the quality of the seeds before release. So that X9 system also used for the quality control stages as well. So where usually people study like 10 markers or 15 markers simultaneously from any given sample. So in a nutshell, different tools are used in different stages. So usually that all breeding projects, they start with the pre-breeding. That stage, microarray are used or sequencing is used. And then once they narrow down the plants of their interest, when it goes to the breeding stage, the people use standard BioTools X9 systems worldwide to screen for the marker assisted selections. Next slide, please. Yeah. So here is one example how that one of the researcher in South Korea, he used our system for marker assisted selection for the QTL identification. So in these projects, they wanted to study about. So this work is published in Agriculture 2021. So this article is from Pusan National University. So in this work, they wanted to identify the QTL that is associated with the rice grain size and grain length and the fragrance of the rice. So they selected a lot of they selected more than 192 SNP. They are known to associated with, and from that they wanted to narrow down to few SNPs. They wanted to identify the few SNPs that is really related to the grain size. So for this project, they use the IFC because for this project, IFC is particularly valuable because they wanted to study 192 targets simultaneously. So this kind of a project cannot be run with a conventional qPCR because they need to study 192 target. If they use a conventional qPCR, that's going to be many years time to complete this kind of a project. So, and it's not only high throughput, it's also provided a cost effective way. That is a main reason they use this technology for their QTL identification work. So this article is published in Agriculture 2021. Next slide, please. Yeah, it is not only used for marker assisted selection and QTL, it can be also used for plant pathogen detection. So this is another example. So when it comes to plant pathogen, there are hundreds of pathogens actually. So if you use a conventional qPCR system, you need to do the reflex testing. What I mean the reflex testing is, so you know that there are hundreds of pathogens. So if you test for one pathogen and then the result stands out negative, that doesn't mean that there is no pathogen. Only that pathogen is not present. So that's what you need to test for the another pathogens continuously till you find out if there is any problem with any diseases. So here you can study simultaneously the multiple target. So you don't need to do reflex testing. For this particular work as a proof of concept, researchers from Forest Research Institute, they wanted to study simultaneously 11 pathogens simultaneously. So because if they use a conventional qPCR, they need to do the reflex testing. So IFC has an advantage because you can study multiple targets simultaneously. They choose particularly this approach to study multiple pathogens simultaneously. Yeah, next slide, please. Yeah. So next. Click next. Yeah. So here, how the seed companies used actually, because seed companies, it is very critical before they release their seeds before sell, they need to do the quality control. First place, they need to do the quality control because they need to check there is a 90%, 95% of the purity in their seeds they are selling. That's what they need to do the testing. So here this slide shows how the seed companies uses. So there is a seed company that use our solution. So they screen more than 200K samples every year. So for this work, they need to screen 20 SNPs from each sample. They need to screen 20 SNPs from every sample. So if they use a conventional technology, they need to do the multiple, they need to run a multiple plate to get the data. So for this scenario, in this particular scenario, so they need to study 20 SNP. So the beauty of that, some SNPs are common among all the plant species they use. So they have they need to study different plant species, but some SNPs are common, some SNPs are different actually. So they were using a different technology that has a pre-spotted, they had a pre-spotted SNPs. So that means that each species, they need to order a different pre-spotted array. So they need to order a pre-spotted array for brinjal separate, and then they separate for the watermelon, separate for the cucumber. So they need to order a different pre-spotted arrays. In spite, they share a common SNPs, and only that few SNPs are different. Yeah, go to next slide, please. So when they heard about our solutions, because our solutions are not pre-spotted, so assays are supplied. So that means that you can omit the assays shared among all the species, you can order only the unique assays, and you can use the same assays when you do the experiment because it is not pre-spotted. So for these projects, so if you use a pre-spotted, you need to stock up hundreds of assays for them actually. For this case, because they are studying a 200K, so they need to stock up the pre-spotted assays in a lot of quantity. So when they use standard BioTools solutions, they don't need to stock up anything. So they can simply use, they don't need to pre-spot, they can buy the IFC and then they can custom on the go, they can customize whatever the assays they want. But in pre-spotted, the problem is if you use a pre-spotted, in any case, you can't change your mind because it's pre-spotted. Only way, let's say if you find out some new SNPs you wanted to add, you need to throw actually the array card. You need to buy a new one actually. But here, you don't need to throw anything. Just simply take out the assays you are not interested and put the new assays. So that is a beauty of the technology here. Yeah. Go to the next one, please. Yeah, go next one. Yeah. So here I'm going to show you some of the examples how it is used in animal health. Apart from plant, it is also used in animal health. So, so even in animal health, it is also for animal breeding. So for this particular work, they wanted to study some of the SNPs associated with HSPB1 genes. So they use, because they need to study the multiple target from these particular things that is associated with heat shock protein. So that is related to the meat quantity actually. So this work is from South Korea. So they published in Archives of Animal Breeding in 2020. So this work also they use our platform because they can study multiple target while it's provide a cost-effective solutions. Go to the next one. Yeah, go next. So when it comes to animal, so animal also has a lot of pathogens. So when it comes to sick, actually, it can be a bacterial infections, or it can be a fungal infection, or it can be a virus or viroid or nematode infections. So when animal is sick, actually, by symptoms, you can't say, simply you can't say, is it infected with the bacteria or by fungi and virus and nematode, with 100% sure actually whether is it infected with a particular pathogen. So it need to be tested. Go to next slide. Yeah, if you use a conventional qPCR, yeah, go to next slide, please. Yeah, if you use a conventional qPCR, the problem with this, again, you may suspect the bacterial disease. You may look for one of the particular pathogens, and then you will be, if the result stands out negative, and then you need to repeat again for the another pathogen, and you need to go on till find out what pathogen is really present. So that's the problem. So that means that you need to do multiple reflex testing until you find out the pathogens of, the pathogen is present or not actually. But when you use this one, so you can put, the target of multiple pathogens in a one IFC. So you can simultaneously study all the pathogen in a one go actually. So you don't need to do reflex testing because on the go, you can study multiple pathogen. For this particular work, so they study about 11 pathogens simultaneously. But it is not limited to 11. As you know that we have you can study up to 96 target simultaneously. So it's not limited to 11, but as a proof of concept, they have shown with 11. But even you can go up to 192 maximum, even without doing multiplexing actually. Even if you want optimize multiplexing, it can be also done actually. Yeah. Go to next slide, please. Yeah. So that's all actually. So I'll be happy to take any questions. So if you have, thank you very much for listening. Yeah. Sure.
Hello. The nematodes are multicellular, and bacteria, we know that these are all very minute and organisms. So how they are going to differentiate basically when they are having two types of infections?
Okay. So thank you for the question. So the test is based on your DNA marker actually. So let's say if you have a nematode pathogen, so that's going to have a very unique primus that is going to amplify the target only from your nematode actually. And for bacteria, similarly, you are going to have a particular marker that is unique to particular pathogens actually. So that's what, it's not a morphological. So it's not, it's based on a DNA marker. So For an example, if the nematode is present, so that's going to give an amplification signal only for the nematode actually. If it is bacteria is present, but for the particular pathogen, it is going to give a signal only for the pathogen. So that's how it's.
Yeah, it's going to be very specific also. I mean, if you are choosing
This, yeah, it is going to be very specific. It depends how well designed your assays or probe or primer actually for particular pathogen. So before any assays is developed, you also need to validate whether it's give any non-specific amplification or not, or it is very specific to your target of interest. Yeah. Yeah. Thank you.
Generally, we are using three different type of instrument for three different type of things: genotyping, expression, as well as SNP genotyping or sequencing. Now, the point is that you are mentioning only one instrument. So that means there are different modules, or I mean, what is the actual thing? There is the one type of workstation, or what is the instrument actually?
So the X9 system at the moment, it can do both genotyping and gene expression. So in both the case, it's a PCR, right? PCR reaction actually. So amplification. So for gene expression, so we use only one probe, like, usually it label with a FAM, or you can use any conventional primer you use actually. For genotyping, so it's usually based on an allele specific PCR. Okay, so there is a chemistry that's called allele specific PCR that can be also done. So for genotyping, if you use a TACMAN probe, it is you can use all the TACMAN assays with our system. So if you use a TACMAN, you can run in real time. But if you use SNP type chemistry or other allele specific chemistry, it can be detect the endpoint. So that means that it can be used for gene expression and genotyping. It is single module actually. It's not because at the end of the day, it's a PCR. So that's what you can run in a single model. But in another few months, you can also do library preparations actually, that will be coming up soon actually. So you can use one system for targeted NGS library preparation, but that will be a new module actually, additional module with the systems.
So you are providing service, or you are providing the instrument?
Yeah, one instrument. Yeah, only one.
What is the present cost of this type of instrument?
So I'm not sure the particular, but definitely we can get back to you if you can. We can talk, and you can leave your email, then we can get back to you on the pricing. Actually.
So it is in India, it is available now?
Yeah, India it is available actually. In India, there are many institutes uses our system, but not the X9 actually. X9 is recently launched. Different institutes, different agriculture universities, they own our system. That's a previous version of our system they own actually. Yeah. Okay, thank you very much. Thank you everyone. Yeah.
Thank you so much, Dr. Ganesh Babu, for talking on the advanced and developed mode of PCR technique for the plant and animal genomic applications. Moving on, we have Dr. Abhimanyu Sarkar, Group Leader, Legume Genetic, NIAB UK, and he will be talking on developing an orphan legume for protein nutritional security in the 21st century. Can we please have Dr. Abhimanyu Sarkar?
Thank you very much for the opportunity to speak. I'll be sharing my screen now. So can you hear me, everybody?
Yes, sir, we
Right.
can hear you. Please go ahead.
Okay, thank you. So yeah, So I'm going to be talking about protein nutritional security and our work on the orphan legume grass pea, which is Lathyrus sativus, also known as kesari, and how we developed resources to breed improved low neurotoxin varieties in this particular legume. So now, as you know, the world faces multiple challenges to feed the growing population. And the world population is projected to increase by nearly a quarter from current levels of around 8 billion to nearly 10 billion in less than 30 years. And most of the addition of this population will be in developing countries. For example, Africa will add more than 1 billion people by 2050 from the current levels. And protein availability is probably the most critical limitation in achieving adequate human nutrition. Now, the current dependence on animal sources of protein is not sustainable in the face of climate change. So, and in the context of the Indian agriculture, India will need to feed probably around 300 million or 30 crore people more by 2050. And climate change is already impacting yield sustainability. For example, last year's heat wave reduced India's wheat production by a few million tons, which impacted the world's availability of wheat, which is also impacted by the war in Ukraine. Other factors that are impacting world food security include soil degradation and the impact of war and other geopolitical factors on nitrogen availability. So the oil prices are increasing. Fertilizer availability is decreasing because of the fertilizer prices increases. Now, legumes offer a solution to some of these challenges, but we need to breed climate smart varieties of various legumes. So we need, when we start a breeding program, we need to figure out what our objectives are to be able to develop these climate smart legumes. One of the most important aspect of any breeding program is that the breeder looks to develop varieties with higher yields. But we also need to have short duration varieties because we want to fit them in agriculture rotations and also reduce input usage, which can be achieved by short duration varieties. This is very important, especially when we talk about water use efficiency. So if you can cut down on one irrigation during the crop cycle, that gives us substantial savings in terms of water usage. But for legumes especially, there is another very important aspect that we need to see, and that is yield stability, even in face of climate or resource challenges. So we are looking for tolerance to abiotic stresses, for example, salinity, drought, heat, or cold flooding. We are also looking for resistance to biotic stresses. We are looking at a good germination or establishment. So these are the traits, of course, and we are looking for high water usage and nutrient usage efficiency. The other thing that we look for in legumes are, of course, quality traits, for example, the protein content, as well as the absence of anti-nutritional factors. So the crop that I'm going to talk about is grass pea, and this is very unique is that in that it can grow when other crops fail. What you're seeing on my screen, and I'm sorry about this particular bar here, I don't know if you can see this, but this is actually a landscape in Ethiopia where the chickpea crop had failed. So the farmers actually just put out grass pea seed, and without any additional irrigation, this is a 7-week-old grass pea crop that germinated on the residual moisture, and now you can see a green field. But if you look closely, there are very few volunteer chickpea that have emerged. But this is actually a grass pea growing in a field that chickpea did not germinate very well. So this crop originated in the Balkan Peninsula in Europe, but it's mainly grown in the Indian subcontinent and in Africa for food, feed, and fodder. And it's a very hardy legume and is often used as the last resort against starvation when there is drought or famine. So, but there is amino acid that's a neurotoxin that is present in all aerial parts of grass pea, which limits its usage. And that's called beta-ODAP. And beta-ODAP can cause neurolathyrism on prolonged consumption when this is the major source of protein. So basically, it causes paralysis of the lower limbs. Now, because of this, grass pea was banned in several countries, including in India. And even now, there are limitations in its usage in the manufactured food chain. So this is a very important limitation for more widespread usage of grass pea, even though it's a very hardy crop grown by farmers. So we wanted to figure out how to overcome this challenge of having grass pea limited by the presence of beta-ODAP. So our solution, or the solution of the breeders, basically has been to breed for low ODAP grass pea, but nobody has achieved zero ODAP grass pea so far. So we first started to look at the existing variation at a global level, and our screen of global germplasm failed to find novel low ODAP grass pea lines in non-improved material. We looked at over 200 plus lines of grass pea from various seed banks, including the USDA, IPK, and the germplasm resource unit at GIC, but we did not get any low ODAP. And we defined low ODAP as something that has got less than 0.1% ODAP in the seed on a dry weight basis. We also characterize the germplasm accessions for protein, starch, and other characters. We also did some field phenotyping for crop phenology and disease resistance over several years. So if we could not find variation naturally, we look to create the variation that would allow us to select for low ODAP. And we achieved this by carrying out EMS mutagenesis, followed by a forward screen for low ODAP mutants. So this is a non-GM approach. And our collaborator at Bench Bio Limited created a population of 3,000 plus M2 families, which we then screened by the Rao method. And Peter Emmerich was a PhD student at that time. He identified 14 low ODAP mutants. And I'll show you the data next. So this is looking at the grass pea ODAP concentration in seeds. And if you look at some of the high ODAP grass peas, there is this European type LS007 that we will come across because we have sequenced the genome of this one. And then there is LSTW11, which was the basis or the parental line for the mutagenized population. When we did our assays, we had pea and chickpea as a control, so that showed a 0 ODAP. We looked at certain existing low ODAP varieties, and there's Mahatheora and Seora from Australia, and Mahatheora is an Indian variety. These came as reliably very low ODAP varieties. There is a Canadian material, LS8246, P24, which was the in yellow, which is the original source of the low ODAP phenotype in most of the Indian low ODAP varieties that had been released till date. But some of the ones like Nirmal and Ratan showed elevated levels of ODAP when we tested it in the laboratory. But the green ones are the levels of ODAP for some of the mutant lines that we had created. And there were at least 4 lines of these mutants that were lower than the lowest available low ODAP variety that had already been released. So this was promising material that we then put into the breeding pipeline. So the other thing that we created were transcriptome resources. We looked at multiple transcriptomes from both high and low ODAP lines. And we ended up with over 2.5 billion RNA-seq paired-end reads. We got these reads from the high ODAP Indian variety that we had got from the USDA. We got LS007, which is a high ODAP European line, and Mahatheora, which is a released low ODAP Indian variety. And we phenotyped them under both normal as well as under drought conditions, and we took tissue samples to do RNA-seq. And this data served as very useful resource for gene model and annotation, as well as for trait discovery in our subsequent work. So we also then sequenced the genome of the grass pea, because this is what was lacking before, and this would allow us not only enable us for to develop markers, but also allow us to sort of look at novel traits and novel alleles. So grass pea is diploid, its 2N is 14, and the line that we sequenced originally is the LS007. Initially we took a whole genome shotgun sequencing approach on the Illumina platform and achieved around 64x coverage on using paired end sequencing of amplification-free libraries. We then did some mate pair libraries to sort of build contigs and scaffold these sequences further. And we initially ended up with assembly that was nearly 8 GB, but had around 2 GB worth of ends. So that's a pretty, not exactly a very good quality library, but we reported this in 2020 in bioRxiv. We then turned around and we tried to increase the length and remove the ends in our assembly using nanopore long read sequencing. And then basically after filtering, we got 35x coverage. And we used software to polish the assembly. This red bean assembly we polished using Minimap and the original Illumina data. So we ended up with around a 6.2 GB assembly that had no ends. And the N50 was around 158 KB for the config. This was, again, a pretty fragmented assembly, around 163,000 contigs. But we were able to identify nearly 34,000 high confidence gene models. And we realized we had captured around 83% of the gene space when we did the BUSCO. This assembly, its annotation has been accepted in Nature Communication in principle. So we are just formatting this, and hopefully this will come out quite soon in the next couple of months. But since then, we have improved the assembly using PacBio sequencing, hi-fi sequencing, and scaffolding. And we have a very good quality assembly now, which is around around 6.1 GB. And this, in that assembly, the BUSCO score is nearly 99%. So we are quite confident that we have captured in our new assembly, which we haven't published yet. We have captured most of the gene space in GRASPy. So that's a very important advance for us. The other thing that we did was to look at the global diversity collection from ICARDA. And we obtained around 435 lines, out of which we resequenced 354 to around 10x coverage. And this is towards developing an eco-tilling platform where we look at the sequence variation that is already available in the global collection at the molecular level. And again, we did paired in sequencing of amplification-free libraries. We also sequenced an additional 30 recombinant inbred lines, which are a cross between the low ODAP, between the LSWT11, which is the Indian type, and a European type, the LS007, whose genome sequence we know now. So using these data sets, we developed markers, SNP markers, identified SNPs and indels, and are validating some of the markers and developing some assays to assist us in our breeding program. So this eco-tilling platform is now in development at the John Innes Center for Allele Discovery, and we hope that this will be available to the broader community in the coming year. So I talked about the GRASPy inbred lines. We have 3 real populations that are useful for trait mapping. And these they are also useful as sort of the entry into our breeding program. So this is a resource that, again, we hope to make available to the wider community. We also developed a GRASPy speed breeding protocol. This is published in Nature Protocols in 2018. This allows us to speed the breeding program and allow us to very rapidly progress generations, which is very useful when preserving diversity between generations is not critical. So, for example, when we are developing inbred or homozygous lines. So just to summarize, we took an orphan crop and we developed genetic and genomic resources that had become, whose lack had become a major bottleneck for crop improvement. This was when I was at the John Innes Center, and we were collaborating with NIAB, which is where I currently am now, but also with ICARDA, ILRI, the Queensland University of Technology, to develop a gene editing platform. BCKV in Bengal, who were our collaborators in the initial phase of our project. Bench Bio, who made the population, the mutagenous population, and Earlham Institute, who helped with sequencing and annotating the GRASP genome. We also added other collaborators as we went along, including the University of Nottingham, who helped us with the nanopore sequencing. We ended up creating a reference genome, transcriptomic resources, low ODAP lines, which now serve as a novel source of the low beta ODAP phenotype that can go into the breeding program. And we also have a gene editing platform now that's nearly ready. We have proof of concept. We will be deploying it for traits of interest fairly soon. And we have 384 genomes that have been resequenced. So marker validation is now in progress. We have identified a lot of markers that would be useful in our breeding program. And we are also looking at rolling out the eco-telling platform for allele discovery. So that's what genomics has enabled us to achieve. And this was a fairly low resource program. So we did not get a lot of funding, but we managed through collaborations to actually achieve what we did. And I would like to thank all our collaborators, including people from India and also from ICARDA, ILRI, the Queensland University of Technology, Crop Trust, and the Templeton World Charitable Fund. Also BBSRC, which is UK's research funding agency for all the funding and support that we have received throughout the project. So, yeah. And I would also like to thank Professor Kole, who has mentored some of our thinking in terms of developing climate smart crops. And that's discussions with him have been very useful. So thank you. And I'm open to questions. And I do apologize for my voice. I'm recovering from.
Thank you so much, Dr. Sarkar, for the excellent presentation. A round of applause for Dr. Abhimanyu Sarkar, everyone. Thank you so much, sir, for the founding presentation and your valuable time. Any questions? Yes, can we pass the microphone? Dr. Sarkar, there is a question. We are taking it up.
Yeah, please.
Hello.
Yeah. I'm sorry, I cannot hear you.
Dr. BC Tripathi is asking a question.
Yes.
Are you able to hear me?
Yes, now I can hear you, sir.
Okay. Which gene you have the mutations so that you get low ODAP concentration?
Okay, so the pathway that was elucidated by Professor SNL Rao's group. So we have some mutations in that, but our recent the upcoming paper in Nature Communications, we have actually identified other genes, other sort of variation in the pathway. The classical pathway for ODAC biosynthesis is we have managed to update that based on our knowledge of the genomics. So these genes, they just exist as numbers right now, but they got to do both with the oxalate branch as well as the BIA synthase branch. And there's quite a bit of biochemistry in the background. I can share with you if you, we haven't actually published some of this work, so that's why I'm a bit reluctant to talk about this, but we have identified genes that if we knock them out, they should be reducing the ODAP levels.
If you identify the gene, then it's very easy to remove the by CRISPR technology now.
Yeah, so that's where we are headed, but it's a bit more complicated than that. The thing is that there is a reason why people, a lot of breeders around the world, they have always got very low ODAP, but they have never been able to get zero ODAP. And the reason for that is there is a, there is a branch in the pathway which allows ODAP to be synthesized even if you knock out the major genes in the pathway. So it's a very complex reaction. We have now identified genes, so we would need to knock out, we think, more than one gene to actually effectively reduce ODAP levels to zero. So even our mutations that we have got, we get them to very low ODAP that are almost undetectable by the Rao method, but are detectable by other methods that we have developed. But we have still not got to zero ODAP, and that's the next challenge.
Thank you.
You're welcome.
Thank you, sir, for the question. Thank you, Dr. Sarkar, for answering the question. We wish you all the luck and success for your future endeavors. Thank you for getting connected.
Thank you very much.
Moving ahead, now let us welcome Dr. Soren Das, Indian Statistical Institute, Barrackpur, India, and he is talking on shelf life of different processed tea, an estimation in relation to their antioxidant potentialities. Give it up for Dr. Soren Das, everyone.
Good afternoon, everybody. I'm talking about the most common and I think it is mass touching beverage, tea. We are lucky that tea doesn't have or need not any promotional advertisement like this. We take tea by practice only, by habit only. Now, as tea is a consumable beverage, then it should have some date of expiry. At least the packaged tea should have. Loose tea, we don't have any guarantee that some destroy when how much it have the shelf life. But on the packaged tea, it should have some best before use, like that tag should be there. As tea, mostly tea we take by habit, it is true, but there are some beneficiary effect from tea, as because tea is a good replacement of an antioxidant elements, supplement of antioxidant elements, or tea mostly refreshing. Apart from refreshing, tea has some health benefit utility also. Day by day, we know that tea not only a simple beverage, but it in ancient China it is used as a medicine also. But then how can you determine the shelf life? That means how much day it have some that best before use, like tag. Before that, down. Okay. Some normally tea is ingested by with just hot water, and it is origin from China, but subsequently it is popularized East Asia and Europe, America, like these. No matter in which season you can take tea, every season you can take tea. Tea mostly we take broadly categorized two types of tea. One is for flavor and another is for just tea, getting some refreshing element. Right. Its benefit go for beyond refreshment. Numerous studies, though these studies doesn't have any medical data, still a different author described it can prevent cardiovascular disease, it can lower sugar, blood sugar, etc., etc. Journal of Preventive Cardiology involved more than 1 lakh adults in China and found those who are regularly drink tea are less likely to develop cardiovascular disease or form of stroke. Different type of tea is there: green tea, black tea, oolong tea, white tea, like these. Green tea is exceptionally high flavonoid content, and that is we know that flavonoid is a polyphenol, and it boost up the heart health by lowering the bad cholesterol and reducing the blood clotting element. Other research found that green tea has possible impact on liver. Mind that, possible impact, though there are no some correlation, direct correlation or medical data, clinical data rather, on these aspects. Still, authors are showing that several results suggest that habitual tea consumption may be considered as an overall health promoting lifestyle behavior. We can remember that in my childhood, people or my parents are always telling, don't take tea, it spoil your liver. But nowadays it is proved that it is not true. In recent years, matcha tea from a green tea, it is grown in popularity. Matcha is very fine, high quality green tea powdered made from the entire leaves of tea bushes grown in the tea shed. Like this, there are some several. If you get 10 cups of regular green tea, in fact, it is a good source of flavonoid. No, it's not reaching. No, okay. Very. White tea, it is least processed tea, may also be good for teeth. Or fluoride, catechin, like this. Oolong tea, partially oxidized, is notable for containing L-theanine. L-theanine can reduce the Alzheimer disease. It is demand that like this. And scientists have found that L-theanine can help prevent congenital disease of Parkinson and Alzheimer. Oolong tea has also high polyphenol, which are linked to lower inflammation, preventing the growth of cancer. I doubt how much it works cancer. Still, there are some reports. Tea production is a very nice, only restricted to some East Asian country or some African countries. China, India, Kenya, and Sri Lanka mainly, they're running, who are producing higher tea like this. But one thing is that Kenya is a small country, but when the export quantity, Kenya is the highest one, almost not highest, second best after China. The graph showing that production of tea from 2006 to 2020 worldwide somehow lowered down in 2020 in China, 2.74 million metric tons to 2.8 million metric tons to 2.74 metric ton in 2019-90 to 2020. This is because of some disturbance in COVID situation like this, I think. And tea production and export like this, graphical representation, world tea production like this, and export is the lower one, lower graph is export. Now, mostly two type of tea export by different countries, Sri Lanka, Kenya, and India, China also, green tea and black tea. We found that black tea export is much more than green tea by different countries. But the prediction from the after 2020 like this, the green tea somehow getting much more popularized because of that. Only that green tea has some excess polyphenol like this, not excess, as because green tea is less processed tea, it has some much more antioxidant value, though there are some hazardous impact also. This is global production of tea per ton, and the lower one is the economic value of the exports. China is running highest, and Kenya is running behind. Sri Lanka and India is fourth. So Indian production is not too much less, but Indian people consume much more than the export. That's why in export, India is fourth. Probably Kenya exports all tea, they are produced. This graph says that temporal world tea production in tons from 1970 to 2020, 10 years interval, it can be found that how much production in tea is increasing from 1970 to 2020. This is and several factors influencing demand for tea, including price, income variable, demographic, such as age, education, occupation, and cultural background. Growing consumption of tea producing countries, driven by population and income growth, has translated in a decrease of exportable production over the past 2 decades. Now coming to the Indian tea. Major tea production in India from 2017 to 2021 and 2022. And the export somehow is lowering from 2019 to 2022. This is because of, I think, some problem is there in 2018, as far as I can remember. Some tea is getting refund from the outside because of their residual fertilizer, like that they found. As 2018 survey, a total of like this 6.37 lakhs hectares are cultivated in the Indian tea production. The northern part of the India is the biggest producer. Northern part means Assam, North Bengal, etc. And Assam Valley and Kachar are the 2 tea producing region. In West Bengal, Duars, Tarai, and Darjeeling. But the major production, exportable tea production produced by the Assam Valley. But in terms of, if you get in terms of money, Darjeeling tea is much more getting the foreign income. India among the first top 5 tea exporters in the world. The types of tea exported through India are black tea, green tea, and oolong tea. White tea is very much costly, but white tea, how it's prepared, do you know? White tea is just a leaf. When the leaf is not come out, does not have the green color, chlorophyll, it doesn't form the chlorophyll. That's why it is tough to collect single leaf from each and every plant. That from that leaf, it is white tea is produced. This is tea history, like this. Now, the beneficial role of tea, actually, if you judge the shelf life of tea, there are several things, how it preserved, how the microbial contamination can be prevented, etc. But this is not my cup of tea. I only concentrate the amount of antioxidant element depleted over the time, only thing. And there are several parameters of tea quality depreciation. But if you concentrate in one single account, that antioxidant element, how it depletes throughout the time. Which are the antioxidant element? Some enzymatics and non-enzymatic also. Catalase, superoxide dismutase, glutamine reductase, like these. Some ROS, RNS scavenger also. Some polyphenol, non-enzymatic polyphenol, carotenoids, vitamin C. Those are the antioxidant elements. Now, the antioxidant and free radical scavenging machinery comprises enzymatic and non-enzymatic system, which regulate biotic and abiotic stress and provided the sustainability. Occurrence of non-enzymatic antioxidant molecules are key component of processed tea and stands for the major health benefit aspects of the tea. Though we said that there are some health benefit aspects in tea, but those health benefit aspects never sustain over the time in the packaged tea. It have some degradation time, degradation value. If you measure the degradation value in a methodical period, in a definite time interval, then you can judge how antioxidant elements are depleting throughout the time. How long you can judge that when the antioxidant element efficiency coming to the zero, then it is useless? No, not only that. You have to restrict in a certain point, 50%, 75%, 50%, 25%. I just like this, that 50%, 75%, 50%, and 25% antioxidant value degradation I just marked. And that is we just named as the shelf life of the tea. Now, tea, 4 types of tea: white tea, green tea, oolong tea, black tea. But all tea is produced from a same plant, Camellia chinensis. But only the difference is the processing mechanism. Plucking of the leaf and weathering, this is the common. White tea doesn't have any steaming or pan frying, drying, rolling like this. Directly drying and white tea is produced. Green tea, somehow steaming, rolling, drying. Mostly processed tea is black tea, rolling, CTC or orthodox, fermentation, oxidation, drying, then packed for the market. That means white tea is the least processing tea, green is more than white, and oolong is more than green, and black is the mostly processed tea. We took packeted material in the same date, same level, same batch processed tea, conventional process, storage material in a room temperature, dry chamber. After 30 days, 60 days, that means 1 month interval, we took the antioxidant data of each tea. But we procured the tea from the market from the same batch, same manufacturing date, like this. And our methodology, we studied parameter is total phenol, flavonoid, proanthocyanidin, tannin, reducing power, ferrous iron chelating, and DPPH, ABTS scavenging activity, hydrogen peroxide, superoxide, singlet oxygen, peroxynitrogen, like this 15 parameters we choose. And the sample preparation is like that: sample powdered, extraction in methanol, then centrifuged like this. And the results is like this. Phenol content, after 120 days, there is a sharp decline in green tea. Remarkable depletion occurred after 210 days in black and 180 days in white. That means phenol content decline in green tea is very faster than the other teas. Whereas flavonoid also higher in green tea, after 90 days drop down much and maintain a gradual depletion. Black and oolong teas, they initially have lesser amount, but degradation after 90 days in oolong tea and 120 days in black tea. That means they are running from the lower side than the green tea, black tea, but they retain their flavonoid content much more than the green tea up to 120 days in black tea. Proanthocyanidin like this and drop after 90 days. Oolong tea have highest initial stage but sharp decline 60 days. Green tea starts from same amount, decrease from 90 days. Black and white tea initially have lower and minimum amount of depreciation noted. Tannin is higher in white tea, almost having similar initial time. After 90 to 50 days, the rate of degradation remarkable. ABTS and DPPH scavenging activity. This scavenging activity, if there are two type of graphs, one is IC50 calculated data is in the right side, and in the left side there is scavenging activity of the ion. Antioxidant ability and their correspondence value have been analyzed. C50 is the IC50 is the expression of concentration of extract which needs scavenge 50% of the radical present in the specific amount of solvent. Free radical scavenging ability has been studied in ABTS and DPPH radical scavenging activity. Percent scavenging activity initially higher in green tea and oolong tea, but depletion rate are almost similar. Fe2 chelation in black tea is similar to the green tea. Reduction of percent chelating ability after 60 days. White tea retained their chelating ability almost same up to 120 days. Okay. Black and green tea shows nearer value. Percent scavenging peroxynitrate assay both decrease after 30 days like this. Superoxide radical scavenging activity and nitric oxide scavenging activity. In both cases, the green tea showing much more stable condition than the oolong and black. White tea also good, showing good. Nitric oxide also starts, radical scavenging starts almost same from black, green and white. Though the oolong starts from the lower one, but oolong retains its radical scavenging activity up to 10 days rather. HOCl radical scavenging activity more or less starts from the same value in all 4 types of tea normally, and uniformly starts to decline after 10 days, right? And black and green tea shows hydrogen peroxide radical scavenging assay at the same as initial time, both sharply decline after 150 days, right? Hydroxyl radical scavenging ability is higher in white tea, lowest in oolong tea. Singlet oxygen, that means O2 radical hunting activity is higher in green tea after a gradual depletion, sharp decline recorded after 300 days. Reducing power occur higher radical hunting value in green tea at the initial point and lose about 50% ability after 60 days. Experimental results of scavenging assay pointed out after 4 to 6 months of preservation, the antioxidant quality of all 3 types of tea gradually reduced, except oolong tea, where upholding radical hunting ability have observed for prolonged time than the 3 types of tea. That means oolong tea is somehow better, having some long life, shelf lifetime. This is a statistical pairwise Pearson correlation coefficient based on the matrix depicts that black tea maximum secondary metabolites were highly correlated with the different antioxidant capacities. Iron chelation and superoxide radical scavenging activity had weak correlation with phenol and flavonoid. In green tea, especially flavonoid and tannin were high. Except DPPH and nitric oxide radical scavenging activity had weak correlation with proanthocyanidin. Similarly, in white tea, in oolong tea, the correlation of the different antioxidant element. To determine the shelf life of 4 type of proposed tea, 15 antioxidant parameter we have chosen. To estimate the shelf life, their antioxidant activity, that is major health benefit attribute have been estimated. The decline percentage of each and individual parameter in after definite interval have calculated like these. Decline percentage is equal to A0 minus AN divided by A0 into 100, where A0 is assessed antioxidant value at the initial time and AN is the after definite interval. The mean declination percentage for 15 studied parameters have considered as predictor value of shelf life. In regression analysis, predictor is dependent variable and the independent variable is the storage time. Best fit curve has been extrapolated, and from these, credible days up for 25% and 50% declination have been calculated. This is the following calculating result. This is the graphical representation of the declination in black, in green, in oolong and white. Processed tea have considerably long shelf life. Processed tea means the black tea is more processed than the other type, 3 type. For its at least moisture content due to involve a series of processing steps during manufacturing. After certain period of storage, different factors like temperature, humidity, microorganism, and tea moisture content causes deterioration of tea quality to a greater extent. Recent report says that compound responsible for the tea health benefit properties like different polyphenol and catechin derivatives are not restored in stable condition during storage. One of catechin derivative, epigallocatechin gallate, decline 1/3 and epicatechin gallate depletes half of their initial content on or before 6 months of storage. Thiaflavin, a potent antioxidant compound of black tea, decreased about 37% and 22% in orthodox and CTC type respectively, and after 12 months of storage. In view of the above, we depicted that phenol content is like this: green, black, white, oolong. Flavonoid content: black, green, white, oolong. Proanthocyanidin: black, oolong, green, white. If we categorize this series, overall initial concentration and activities were almost maintained up to 90 to 120 days, and thereafter declination starts. After 180 days, rapid declination starts, and beyond 330 days, 60 to 75% of the initial activity decline, according to our result. The predicted shelf life period, maximum days of oolong tea type, could be inferred about the above order, as much of the antioxidant component least degrade throughout the storage time. It is initially noted, although black tea contains lower antioxidant value than the green tea, it is less affected by the storage period than the green tea. It is hard to quantify the total antioxidant activity based on single active component. Therefore, total form of antioxidant activity has been estimated along the individual radical scavenging property. It is reflected that after a certain time of storage, metal chelating activity of black and green tea drastically declined, whereas the same is steadily maintained by both semi-fermented oolong tea and slightly fermented white tea.
Thank you very much. If any question is there.
Thank you so much, Dr. Das. Any questions? Should we move ahead? Okay. Thank you so very much, sir, for the commendable presentation. Round of applause for Dr. Soren Das, everyone. The presentation itself enhanced the recalling value of the term tea, that everyone in the house are in the urge of taking some tea or coffee, right? And especially the students sitting over there, they are in the need of some antioxidants because of back-to-back sessions. So we will definitely go for some tea and coffee networking session. But before that, we have more two speakers. For now, let's welcome Dr. Sagarika Parida, Associate Professor, School of Applied Science, Centurion University, Bhubaneswar. A big round of applause for her. And she is talking on exploring the impact of vermicomposting on soil health and growth of Triticum aestivum L on the organic farming system. We welcome you, ma'am.
Good afternoon. Good afternoon, one and all present here. I am Dr. Sagarika Parida, Associate Professor, Department of Botany, School of Applied Sciences, Centurion University of Technology and Management, Bhubaneswar campus. Before going to this topic, exploring the impact of vermicomposting on soil health and growth of wheat crop, I would like to say a few lines. The motivation behind this work. Actually, one of my cousins was suffering from cancer and advised by the doctors from Tata Memorial Hospitals that he should take wheat grass juice early in the morning. And it was a hectic schedule for his son to grow and to get the leaf every day. And that motivates me to do this work. Next slide, please. Here. Okay, thank you. So in this experiment, we have grown the wheat seeds in organic farming system. We know that organic farming system, soil health is maintained by organic farming system, and soil health is defined as the capacity of the soil functions, the capacity of soil to function as a living ecosystem, and it can be measured by its nutrient contents, its water holding capacity, and also the microorganisms present in it. And this, we know that the soil is a living ecosystem, and about 87% of the soil that is sustained sustained all the life forms on this earth, about 39 inches of the soil that sustains the life forms from microorganisms to the human system, and also plants, as certain animals. Every living organism sustained on the 39 inches of the deep soil, soil. Then agricultural soil are invariable deficient or imbalance in one or more essential nutrients, mostly because of some agricultural practices resulting in reducing the crop yield. So these are some of the agronomic practices. Agronomic practices like tillage. Tillage is the preparation of the seedbed, and then use of chemical fertilizers and use of pesticides and monocropping. And these pictures I have collected from the net sources, and these are the agronomic practices that degrade the soil. And other responsible for factors for soil degradation, there are two factors: natural causes and man-made causes. Natural causes, we know that landslides, hurricanes and volcanic eruptions. Then man-made causes, urbanization, domestic waste, industrial waste, mining and radioactive emissions, dam construction and agricultural waste. So agricultural practices to maintain the soil health and enhancing yield, there are different factors. These are the factors here: reduction in chemical fertilizer use, pesticide management, integrated nutrient management, then foliar fortification, proper tillage operations, mulching. Mulching is the covering of the soil. Then residue management, nurturing the rhizospheric region, and nano carrier based fertilizer plant growth, boosting nanoparticles, and use of organic fertilizers or enhancing organic farming system. So we know that organic farming system has positive impact on environment and soil ecosystem, and thereby its positive impact also on human health. But organic farming system is criticized for low yielding, as a low yielding system. In this study, vermicompost and cow dung compost was applied to observe its impact on growth of the wheat crop under pot cultivation experiment. So these are my objectives: impact of vermicompost and cow dung compost on soil nutrient status and wheat crop growth, and its impact on wheat crop nutrient status. Then we have collected Pusa DBW 303 Karan Vaishnavi variety, and vermicompost is collected from the vermicomposting production unit CUTM Bhubaneswar campus, and a field experiment with wheat cultivars in organic condition was performed in pot cultivation method in February 2022. Then we have taken pot containing normal soil as control, then soil and cow dung in 2:1 proportion, soil and vermicompost in 1:1, and then soil and vermicompost in 2:2, soil in vermicompost in 4:1 ratio. And after that, we evaluate the vegetative crop growth in intervals of 7 days, 15 days, 25 days, and 35 days after sowing. These are the methodology for collecting the soil samples. So we have first of all cleaned the soil surface and then dig a hole in V shape for soil analysis by XRF technology, and about 15 to 20 centimeter dig we have done in a V-shaped manner, and 2 to 2.5 centimeter thick slice of soil was collected from each side of the exposed site of the V-shaped structure, and then collection of soil samples from 8 to 10 places. The soil samples were collected from different places of this campus from the medicinal plant garden, and then all the samples were mixed well. Then after that, 2 grams of soil samples were sent to ATC laboratory for XRF analysis. Then these soil samples were filled with different proportion as suggested earlier, in different proportion in different pots, and all the experiments were done thrice. Then, pre-soaked seeds were sown in row manner. Then raising of seedlings, the wheat seeds were pre-soaked overnight And it was shown in the soil filled with pods in row manner. And for soil nutrition analysis, XRF analysis was done, and growth of the seedlings were measured at regular intervals. So we have studied this soil nutrition, then wheat crop growth, then nutrient contents in wheat leaf. These are the different nutrients present in soil samples in percentage. That is aluminum oxide, dioxide, then silicon oxide, and phosphate pentoxide. So these are the different nutrients present in the soil samples. And out of these, soil and vermicompost in 2:1 ratio proved to be the best for the crop growth, wheat crop growth. And these are the some nutritional factor, nutritional composition or nutritional content at ppm level. And nutritional composition in soil and vermicompost showed optimum growth in comparison to other soil composition, also in soil and cow dung compost. And then these are the wheat seedlings. After the first slide, it is after 7 days, and then second one, it is after 15 days. And these are soil vermicompost 1:1 ratio, then soil control, and 2:1 ratio soil is to vermicompost, and different proportion of soil, 4:1 soil is to vermicompost and soil cow dung. And this last picture is that soil vermicompost 2:1 after 35 days of sowing. So these are the growth of the wheat plants, wheat grass, after 15 days of sowing and after 25 days of sowing. Average leaf length is maximum in soil vermicompost 2:1 ratio, and then followed by soil and compost. These are graphical representation of effect of vermicompost and cow dung compost on wheat crop growth in 15 days after sowing. And in table 3, wheat crop growth in control and soil vermicompost after 30, it shows 35 after 35 days. In soil control, the average plant height is less in comparison to soil and vermicompost. And also average leaf length is also less than the soil vermicompost in 2:1 ratio. So these are the graphical representation. This is nutrient contents in wheat leaf. After 15 days, we have collected the wheat leaf, and then we analyze it by XRF analysis. And these are the nutritional composition after 15 days. These are the graphs, XRF graphs, showing different nutrients present in the leaf. And these are the elements in leaves from pots, soil and control, and soil and vermicompost in 2:1 ratio after 15 days of sowing. So for wheat crop, application of vermicompost in soil showed better result on crop growth than cow dung compost and other treated soil composition. Soil and vermicompost may have optimum nutrient content for better vegetative growth. Apart from this study, now we are also in continuing that zinc oxide foliar fortification to raise the zinc content of the wheat grass, so that it can be used as the immunity booster, because that wheat grass juice is also known as living juice, and also it is known as the green blood. And because it has antioxidant activities, and also it eliminates the toxic elements from the body, and also it contains several nutrients like several vitamins like vitamin A, vitamin B, B1 to B6, B8, B12, and vitamin C, vitamin E, and vitamin K. So definitely, if we increase the zinc content, now we have also got some results. I have not included here. We have enhanced the zinc content in the wheat grass, and we can consume the wheat grass juice or the green blood or the living juice as a supplementary food to boost our immunity. Thank you. Any question? Yes. Yes. Yes, sir. Yes, sir. For 2 kg of soil. No, sir. I think it depends upon variety, because some variety does not show that proper growth in only soil. Also, it does not show any proper growth. It depends upon variety. And 2:1, sir, soil is 2 kg, and vermicompost is 1. Sir, cow dung is also 2:1, and vermicompost in different proportion, 1:1, 1 kg, 1. Sir, actually, we are generally using, yes, sir. Sir, we are using that cow dung. Generally, we are using cow dung, but here we are we have produced the here we have a unit, vermicompost unit, and we are producing that, manufacturing that vermicompost using that plant debris and other organic products, and we want to see whether this vermicompost has any positive response towards this germination of the wheat seedling and grass, sweet grass. Okay. Yes, sir.
Yeah, so first of all, thanks for a nice presentation, madam. When you shared that something is good for your soil with the vermicompost as compared to soil and cow dung. So when you are saying something is better, so in terms of statistics, I think it's important.
Yes, sir, we have to include the statistical significance. We have to look that we are in the process of other parameters study for other parameters. We have to include that also.
And what is the basis of that X-ray fluorescence analysis? I mean, how does it work? XRF technology that you said?
Yes, sir. In XRF technology, if we can measure all the nutrients present in any substance, in also leaf also you can measure, and also XRF by XRF you can taste the soil samples also.
No, what is the principle for that? I mean, how does it work?
Sir, that I cannot answer. Okay, thank you.
Thank you so much, ma'am, for the excellent presentation, and also for sharing the story behind your work, the real life inspiration behind your work. We wish you luck, ma'am. Thank you so very much for the excellent presentation. Moving ahead, now let's welcome Dr. Raghunath Satpathy, Assistant Professor of Biotechnology, GM University, Sambalpur, and he is talking on in silico prediction of hop genes and pathways related to osmotic tolerance in Arabidopsis thaliana. He is with us virtually. Let's welcome him.
Good evening. Am I audible?
Yes, very good evening, sir. You are very much audible. Please go ahead.
Yeah. Let me share my screen. Is it visible to all? Is it visible?
Yes, sir. It is visible now.
Okay. So good evening, one and all. So I am going to talk about it's in silico prediction of the hop genes and pathways related to osmotic tolerance in Arabidopsis thaliana. before going to this prediction, let us discuss about some few of the thing like concept of the gene expression. As already we know, so DNA codes for the protein through this RNA, and basically this coding RNA is mRNA, and that generates or that expresses this particular protein. And
Sir,
protein
just
is
one
the,
second. Sorry for the interruption.
Yeah.
I request you to kindly share the full screen. Please tap on the full screen.
okay. Is it okay now?
Yes, go ahead, sir. Thank you.
Yeah. So this is the coding RNA that gives rise to protein, which is the key player of any kind of process. So the gene expression pattern, if you will see, some genes are differentially expressed because there are different cell types, different environmental conditions, developmental phases, disease states or stress, whatever we say. So there are many conditions. These gene expression are differentially expressed both in animals and plants and any kind of living things. And also, the gene expression is primarily regulated at the level of transcription, and this copy number of mRNA is very important. For example, if it is a highly expressed gene, they might have about 10,000 of mRNA. Sometimes it is very less also. So to detect or to find out these gene expression things, we are basically using the technique microarray, which is a method for high throughput gene expression profiling involving the hybridization of mRNA to an array of complementary cDNA probes. And this hybridization generates the intensity of a particular probe, labeled probe, so that expression level that can be computed. This is the very basic technique what we are using in the microarray. There will be a control, there will be experimental, or you can say it's in a stress condition or any treatment, what you can say, treated cell. So this cDNA is to be extracted, mRNA is to be extracted, and it is converted to cDNA and label thing, so that after the hybridization, we can get some idea about the expression of the gene, which are up-regulated, which are down-regulated, which are constitutively expressed, all these things. So this is the basic thing. What is this application? We can infer many kind of result from this microarray experiment. For example, cell-specific gene expression, what I already discussed. Then regulation of gene, how the environmental conditions impact on this expression level of this particular gene. Then elucidation of this metabolic pathways, what particular pathways they follow so that this particular gene is expressed. And there is a server that is called NCBI GEO. So Gene Expression Omnibus, it is a public repository that actually stores this microarray experimental data, including the next generation sequencing and other forms. These geo datasets can be used because it provides the high throughput functional genomic data. It supports complete and well annotated data. It is freely available for any users. You can download and you can study the expression pattern of this particular gene of interest. So there is a tool available in this NCBI GEO. It's GEO2R. It is an interactive web tool that allows the users to compare two or more groups of samples in this GEO series. Okay, there is another database is known as string database. So string is a database of known and predicted protein-protein interactions. These interactions include physical or functional associations. You can predict. It's just a screenshot to show you how these genes are. Then what is a hop gene basically? So hop gene you can see here. It's basically the gene having many interactions with other genes. So since we have thousands of gene interaction we are studying through microarray study, it is better to calculate to predict these hop genes. So because it plays the essential role in gene regulation and a particular biological process. So in this present analysis, we have attempted to identify some key genes associated with the osmotic tolerance of this Arabidopsis thaliana. So for that, we have taken, let me directly go to, so these are certain, what I can say, some of the plant genes that is related to, it's already reported to this osmotic stress, that is related to osmotic stress. This particular proteins are expressed basically. So in this case, so we basically, we took this, we retrieved the data of a microarray expression profile from NCBI GEO. This is the accession number of this particular data set of Arabidopsis thaliana, which was available from April 2020. And the differentially expressed gene we calculated by using GO2R. There is certain condition we kept, like the adjusted p-value. It is available in this expression profile, and log fold change it should be 1. So this condition was kept. So those who qualify this particular condition, this is considered by differential expressed genes. We can change the value also. For the standard things, we have taken that particular thing. Then the protein-protein network constructions, we selected all this particular gene, whatever obtained from this differential expressed gene. Then we took these particular genes were fed to the string server. And there also we selected some score, like it's greater than 0.7. For the to avoid the distance related, I mean, genes. So we are having very good score. I mean, good connection genes will obtain. Then these hop genes from this particular result, we have used a another tool, Cytoscape, along with a plugin Cytohuba. So that produces this thing. So from the result we got initially, after first screening, 297 differential genes. After screening of the network from the PPI, I mean in string server, we found there are 98 genes and 278 edges of the particular things. And this was fed to this Cytoscape, and we obtained there are 6 genes. Particularly as a hop genes. These are the thing. So these are the relationship between these particular interactions of these particular genes. And if we see the, we have annotated what is the function of this particular gene. These are having different functions, like it's for example, NUD T7 is a Nordic hydrolase, is responsible for hydrolysis of these phosphates. Then CBP60G, it is a calamidolin binding protein. Then WRKY3, it is it is already reported in its function as a defense response. Then you are having again calamidolin like proteins. Then syntaxins are also there. There is vesicle trafficking protein. Then leucine rich repeat receptors. There is also that is called SOBIR1. It is a this is a counter player or inhibitor of this particular kinase. So in this way, these 6 I mean genes or the was found as this. Were found as the hop genes for these things. So these are related to the osmotic stress. So presently the work is going on to calculate their physical interaction by protein-protein docking and how they actually interacting to each other, so that we can reconstruct the pathway for this protein content. So these are some of the reference. And thank you for this thing.
Thank you so much, sir. Can we take some questions? Yes, please.
Yes, yes.
Hello. Yeah. Can you hear me?
Yeah, yeah.
Okay, thank you. Okay, so in the microarray that you did, normally you did the microarray to see the down and up expression of a lot of genes over there.
Yes.
So for the validation, normally it is followed with some QRT-PCR experiment also. Because when we do microarray, what I know, you need to do some RT-PCR for the confirmation of those kind of genes to be up regulated or down regulated.
Actually, it is an in silico work. We have already taken the data set that is already available in the database of these things. We have not verified. Okay, that is the thing.
Okay, thank you.
It's just to check what are these hop genes, and currently we are doing the protein-protein docking. This thing is going on. How physically they interact.
Okay, thank you so much.
Thank you, sir.
Thank you, Dr. Satpathy, for the commendable presentation. This was the last presentation of the day. Here we come to the end of day 1. I request all of you to kindly join in the inaugural session and follow the cultural session. Thank you so much, everyone, for your patience hearing. Thank you, all the speakers, and especially the students present over here. See you in the cultural session. Thank you so much.
हो गया